Hello, everybody. Welcome to Advancing Gene and Cell Therapies in South Africa. My name is Ken Cornetta, and I'll be co-moderating this with Dr. Malangu. The goal of the symposium is for US and South African speakers with experience in clinical development to share their experiences and insights on factors to consider for successful initiation of clinical trials of gene and cell therapies in South Africa. Stimulating such additional clinical research is an important step in expanding availability and access to these therapies for patients in need. At the end of each session, when we'll have two speakers within each session, we will then open it up for a panel Q&A session. We encourage you to type questions into the Zoom chat feature throughout the presentations. And you may continue to submit questions throughout the panel discussion as well. So to kick it off, I'm going to make a short presentation just to maybe help orient folks and give a little bit more background. So to start out, this meeting has been co-sponsored by the American Society of Gene and Cell Therapy. We're an organization now that's been established about 25 years ago. And I think it was important in the mission statement to sort of look and say what our goals as a society has been. And that's, again, to advance knowledge and awareness, but also to really help alleviate human disease. And that is really a main mission of the society and part of the reason we developed this program. And sort of, again, maybe just for folks who may not be familiar that much with gene therapy in particular, just wanted this slide to say that the clinical protocols and trials that we're talking about in this area really look at somatic cell repair, and we're treating the patient. The kind of things we're looking at now do not try to change the germline. Generally, there's been consensus that that would not be ethical at this time. So we're not really trying to change the gene pool, but we're trying to treat individuals. A lot of the things that you'll hear talked about today will be around viral gene transfer. And this is just a simple diagram. The most common applications now in gene therapy have been looking to use viruses to introduce material that may be potentially therapeutic. And the MLV is a very simple murine virus. And if you can see, what we've tried to do is actually take out the viral genes, put in a therapeutic gene, and then use the virus basically to package it to deliver that gene to the cell and then correct the cell's defect. There are a number of ways. Not everyone uses viruses. Plasmids may be used. They aren't expensive, but they're not very efficient. And so you need to have generally direct contact with the area that you want to transduce. But there's a number of viruses that are in currently. The retrovirus and lentiviruses are very common, and AAV is another very common virus that has been modified to deliver genetic material. But if somebody has a virus, they've been working on trying to use it in a gene therapy setting. And we do viruses because they have some certain advantages. For example, you'll hear about lentiviruses today, and they integrate. So trying to transduce cells such as bone marrow cells or T cells, where you expect that cell to grow, to multiply within the patient to try to treat that disease, we want to get integration so all the daughter cells will be treated. AAV has advantages in that it's relatively able to be infused intravenously. It doesn't integrate, but it will infect and then stay for a significant period of time within cells that are not dividing. So for liver, muscles, and other organs, including the CNS, it's being currently utilized in investigations. Now, I just put this slide here, sorry, to give folks an idea of what kind of gene therapy trials are ongoing. The majority are for cancer, and that's the yellow part of the pie. As you can see, monogenic diseases, cardiovascular diseases, infections, neurologic diseases, basically many different investigators are looking to treat a whole variety of diseases with gene therapy approaches. And some numbers are putting here. And not only are these clinical trials, but these are actually now being approved in various parts of the world. And there's estimates by 2030 that there'll be over 60 approved cell and gene therapy products. So it's really starting now to be in the clinical realm. Part of the challenge is when we look at where these gene therapies are being developed, it's limited in area. And so looking, this is from clinicaltrials.gov, you can see that in a variety of different countries, there's relatively few clinical trials open. And it's been concentrated more in Europe, the US, and in China. And if you sort of look not only where the geographic distribution is, but also the population distributions, there's clearly a real shift in where these are available. And there are parts of the world, Asia and Africa, where there's relatively little access to these types of therapies. And then the other challenge for these therapies, particularly gene therapies, is their expense. And this is an issue even in the US, is because some of them have really probably actually put on price tags that may have taken them out of the market. For example, Glybera, which had $1 million, and then this other newer treatment was listed as the most expensive drug ever. So these are health disparities that are occurring everywhere. And it's just become a very challenging topic for folks to try to address. So I know actually in Indiana, for some of the newer therapies, for example, CAR therapies, the reimbursement by our health system is very, very low. And the insurance reimbursement makes it almost impractical to try to do things. So we're looking at how do we actually bring that technology in more locally and do it so that we can actually treat the patients. It's a pretty complicated endeavor. If we're going to develop therapies, we need basic scientists, we need clinicians to understand the diseases that we're going to treat. And I think you're going to hear about some of that today by some of our speakers. But then actually, how do we make the material? We can have gene therapy vectors, but genes only work in cells. So how do we do the cellular therapy part? These can be somewhat expensive, but how can we try to also then share amongst ourselves, since it's challenging sometimes for one institution to do all of this? And so this is a model that, again, hopefully we can start to see some things in our talks today to maybe try to do some homegrown development of gene and cell therapies. So the talk today came really from the ASGCT strategic plan. And in 2020, they formed a new committee called the Global Outreach Committee. And it was really to say, we're actively working in the US and we have many members throughout the world. But how do we partner with other professional societies to try to make sure that educational content and additional collaborations develop to foster equitable access to gene therapy, particularly in lower and middle income countries? And the society does have some things on its website that can provide education around gene and cell therapies. They have actually some what they call patient journey units that can show examples of various diseases that can be treated with gene therapy. You can actually find open clinical trials on the Clinical Trials Finder. And for folks who are working in the research area, there's a new investigator center that has a number of educational resources. The other thing that the society did in this past year was actually to reduce the dues to join the society. If you're a lower middle income country, it's a 75% reduction. And if you're a lower income country, it's a 90% reduction in the cost of dues to be able to be members of the society. And virtual symposiums like the one we're at today is here. We actually had one with Brazil back in December, and we're planning for another one with folks that we're collaborating with in India in October. We've got a large number of really dedicated folks who are on the committee from a variety of different countries working towards this. And I wanted to really special thanks to Johnny Malangu, who has really been helping develop this and is now a member of the committee. And as we get started, I want to thank Samantha Kay and Betsy Foss Campbell and other folks at ASGCT who's been able to put this together. So I'll just put this up for a second. And I think it's time now that we move towards our first speaker. And it's my pleasure to introduce Johnny Malangu. And he's a professor in South Africa and the Purcell Professor of Hematology and head of the School of Pathology at the University of Witwatersrand and also the National Health Laboratory Services. And we're good. It's our, I think I'm excited to hear his talk today on gene transfer of genetic diseases. And Dr. Malangu, welcome.
Dr. Malangu, you may be on mute.
Great, and thank you, Ken, and apologies for that glitch. Are you able to hear me now?
Yes, we can.
Excellent. And good morning, good afternoon, colleagues. An absolute pleasure to see you all in this ASGCT symposium webinar. We all decided that in fact it's time to bring Africa into this bus. And my job today is not so much to talk about probably what most of the attendees here know, but to allude to some of the progress that have been made in some areas of gene therapy. It is a pity that I can't talk about personally what I'm doing at the moment. We're involved in a number of gene therapy programs, and some of that data, in fact, is under consideration either for publication or for presentation in a congress next month. So I will not be sharing some of that data as yet, but hopefully in the near future. I was asked to talk about gene transfer for genetic diseases. These are my disclosures reflecting largely my interface with clinical trial sponsors. Just to remind you, and I think Dr. Koneta has already mentioned that there are a number of approaches in gene therapy. At its basic level, there are two approaches for curing genetic diseases. The one is the in vivo and the other one is the ex vivo gene therapy. The in vivo gene therapy, one would be required to identify the gene of interest and then identify a vector. And there are two types of vectors that we use. These are viral vectors as well as non-viral vectors. And I will not be talking much about the non-viral vectors, and perhaps we can talk about later because they are very early in their development as tools for gene therapy. The second approach is ex vivo gene therapy, in which the cells from the individual that suffers from the disease are taken out. They are manipulated and be given the missing gene, which then gets infused. The cells are infused into that individual with the understanding that the cells together with the genes will then be able to provide the missing protein, for example, in the case of diseases where there is a missing protein. If one looks at the vectors in general, there's a very large number of these vectors that are currently being developed. And I would say that the majority of them are viral vectors. As you can see in this schema, the majority of the vectors are either adenoviruses or lentiviruses or adeno-associated viruses. In fact, only a small proportion of these are non-viral vectors. And if one looks at the viral vectors in closer detail, the majority of those that are used for gene therapy are adenoviruses, followed by adeno-associated viruses, and then of course a small number of lentiviruses. By the way, that is a group that I think is growing very rapidly, and probably in the next decade or so will overtake the use of adeno-associated viruses. And we'll hear about some of the shortcomings of the adeno-associated viruses. Nevertheless, they are the ones that are favored and being used currently in many of the gene therapy programs. Just to give you an indication to what extent are adeno-associated viruses being used in currently approved gene therapy programs. As you know, we currently have 5 gene therapies that are approved by the FDA and the European Medicine Agency. 3 of these are using adeno-associated viruses. And of course, the CAR T-cell are using lentivirus and gamma retroviruses. And in fact, one then begin to understand the importance of the viruses as vectors for gene therapy in the context of what we're doing at the moment. And this is a schema representing the process that we normally follow in gene therapy. One identifies the gene of interest, would then package it into a vector. The transgene, together with the vector, of course, is then given to the individual who is deficient in a particular protein. And I must admit, that bit then becomes a black box. And what we expect is ultimately the production of a protein which initially was missing in that individual. There are several, of course, intracellular steps that happen in an individual, some of which we understand very well. I beg your pardon. We do understand, of course, that the vector will have to find its way to the target cell, will have to dock onto the target cell, get into the cell, get into the nuclear space, release the gene of interest, and of course express the protein, which then gets measured often in blood. That black box, in fact, can be generalized using this cartoon. And some of us are particularly interested in what actually happens inside the cell. The vector ultimately docks onto the target tissue or organ of interest through the receptor. There is usually a receptor-mediated internalization of the vector with the formation of the endosome. The endosome is disrupted with the release of the nucleic acid, which then gets transcribed and then translated into the protein that is released into circulation. And I must emphasize that, in fact, whilst we may not understand the nitty-gritty of what happens at cellular level, that has not stopped us actually from conducting clinical trials in humans. There is a very large number of clinical trials currently being conducted in humans. In the phase 1 and the phase 2, just over 3,000 clinical trials being conducted. And of course, a smaller number of those are entering phase 3, and some of them, in fact, are actually in phase 4, with a fairly small number of individuals, just over 174 individuals in the phase 3, phase 4 space. But the bulk of what we are dealing with and hope to deal with in the near future are clinical trials that are entering into phase 1, phase 2 studies, which are extremely exciting, as you can tell, over 3,000 of those. I will not bore you with exactly the kind of clinical trials that are currently being conducted. Suffice to say that some of them are meant to cure cancer. Some of them are for genetic diseases, including those genetic diseases with which are amenable to genetic cure, and many others miscellaneously. What I want to focus perhaps in the next part of this presentation is a small subset of the genetic diseases in which there is failure of production of proteins with hemostatic effect, the so-called hemophilia A and hemophilia B. Just to remind you that at its basic science definition, hemophilia is defined as failure of thrombin generation. The failure of thrombin generation, of course, will result in the absence of clot, and hence these individuals will bleed. The failure of thrombin generation is due to either deficiency of factor 9 or factor 8 in the presence of the natural inhibitors that one would expect in the body. Phenotypically, the individuals will present with all sorts of bleeding, but the hallmark is usually intra-articular bleed. In a subset of patients, there is an element of chronicity that evolves in which you get chronic synovitis. But in all patients, whether you've had one bleed or 1,000 bleeds, ultimately you get the destruction and dysfunction of joint, as shown here by this X-ray showing almost absence of joint space. The good news, of course, is that this sequence of events can be stopped by replacement therapy, which we know is the standard of care. And in the last several decades, we have seen this unprecedented landscape in the evolution of the therapeutic tools for the treatment of hemophilia. Starting with blood and blood products, these culminated in 1964 by the use of cryoprecipitate when Judith Poole actually formulated the first cryoprecipitate. We have seen, of course, since then a move away from blood and blood products to clotting factor concentrate, which were initially plasma derived and subsequently recombinant with the advent of DNA technology in the 1970s, and of course, more recently, the extended half-life products. Replacement therapy does not come without a baggage. There are a number of challenges associated with replacement therapies, and I've listed 3 here. The one is immunogenicity against the protein that is being produced or replaced. The development of antibodies is the single important challenge in replacement therapy. And of course, all of these proteins have to be given intravenously. The burden of treating individuals with the deficiency is huge, and that often compromises adherence. And finally, because you are dealing with a protein that is not continuously produced in the body, you get peaks and troughs which result in non-hemostatic effects, resulting in breakthrough bleeds and inadequate factor levels, leading to, of course, breakthrough bleeds and subclinical bleeds. It is in that context that gene therapy for the hemophilias, in fact, was seen as a future solution, and it is meant to address these challenges. And I've given you here a summary of where we are with gene therapy in hemophilia. Let's start off with hemophilia A. We've been using AAV-mediated gene therapies, and I've listed here at least the 7 studies that are currently ongoing or have been ongoing. Perhaps the critical elements here, not to confuse you with the many details in this table, is the fact that, in fact, we've used 2 types of genes. The genes that can deliver the traditional factor 9 and the gene that delivers a hyperfunctional factor 9, the so-called factor 9 PADWA. And that is obviously important in the context of accelerating the therapy for patients with hemophilia. Perhaps another point to make here is that all of these studies use adeno-associated viruses. The majority of these viruses are either AAV5 serotype or AAV8 serotype, with some of the viruses that are manufactured, the novel viruses, adeno-associated transgene that are manufactured in the lab. What about the dosing? The dosing has been fairly variable, ranging from 2E11 to as high as 2E13 in some of the studies. And of course, these studies are ongoing. One cannot comment as to which of the doses are actually more effective, or at least more efficacious in the context of delivering the therapy that one needs. If we move on to the kind of population the studies in hemophilia B have all been restricted to date to patients who are adults, in other words, 18 years or older, as you can see here. And in fact, all of these studies have got preliminary data that shows that the therapeutic genes are actually have got assumed efficacy and safety profiles. And some of those, in fact, are holding onto the ongoing studies. Perhaps the disappointing bit with hemophilia B is only one study that has entered into the phase 3 studies. All of the other studies are ongoing. Of course, the patients will be followed for a long time, but they're ongoing pre-phase 3 studies that one is watching carefully to see where we are, despite the fact, of course, that hemophilia B was the very first gene therapy that made a lot of promises for our patients with hemophilia. If we switch over to hemophilia A, also AAV-mediated gene transfer has been explored extensively. As you can see that the gene delivered is usually the B domain deleted factor 8. The vector being used is usually the AAV5 or the AAV6. The dosing regimen has been explored quite extensively, and we seem to be settling in the region of 6E13, 4E13 here, vector genomes per kilogram. And of course, lower doses have been used, and perhaps we'll touch on that later when I show you the other data. The age is also adults. These have not been tried in pediatric and adolescent patients. Preliminary data is shown in the majority of these studies that the safety profile and the efficacy profile is acceptable for these studies to be allowed to proceed. What is particularly exciting with hemophilia A gene therapy is the fact that, in fact, almost all of them have entered into the phase 3 programs of their development. And most of us are extremely excited about the fact that hemophilia A actually affects the majority of our patients, and therefore it would seem that this will advantage some of the patients, at least, who may take advantage of the advances in gene therapy in hemophilia A. And of course, I just want to share with you what is in the public domain. I can't talk about what is not in the public domain. There's been a number of high-profile publications out of the hemophilia therapy, gene therapy programs. And I'm showing you here the 3 hemophilia B studies that were published in the NEJM and Blood. As you know, these are fairly prestigious journals. And there is one study that has been published in the NEJM on hemophilia A. I just want to summarize for those who have not had an opportunity to read all of these papers, at least together, the data from these papers. And I've taken the liberty to put them in tabular form, and I will take you through some of the thinking behind it. Perhaps the first thing to summarize here is that all of these studies are conducted in adults, and in fact, in adults, some of whom may well be over the age of 50 and have set arthropathy, and yet they felt the need for them to be part of the evolution of these therapies. Secondly, that the majority of the patients, of course, are between 30 and 50 years. These are usually economically and productive individuals. It is actually very fulfilling to see that these are the individuals that have chosen to participate in the studies. And if one looks at the total number of patients, it's a small number. It's only in total for the 4 studies, 43 patients. I've no doubt in my mind that that number will start to grow as soon as some of these gene therapy programs, in fact, are commercialized and made available widely. You'll see that it would seem for most of these studies, the number of participants were restricted to at least 10 patients. And I've no doubt that that was guidance given by the regulatory authorities. What were the efficacy outcomes? If one looks at the expression of factor 9 in the 3 studies, you'll see that the lowest level was 1.1% to as high as 60% in those patients that had received the factor 9 gene therapy. In the case of the factor 8 gene therapy, the lowest was 19%, and the highest mean was 237. Needless to say that these are means. The range, of course, the ranges are very, very wide. What about the other efficacy outcomes? If one looks at the bleed rates, in fact, gene therapy delivered exactly what we wanted. The absence of bleeds in the majority of our patients. If you look at the bleed rates in post-gene therapy across the programs in hemophilia A and B gene therapies, you can see that there is a significant reduction of over 70% across the board, with some of the studies delivering 96% reduction in bleed rate. If one looks at the utilization of replacement therapy, gene therapy actually puts out of business replacement therapy. As you can see here, that majority of the patients that have undergone gene therapy actually do not bleed, and therefore they have no need for replacement therapy. The remaining 10% are in my mind, it's quite clear, those are individuals probably that have had traumatic events for which they need replacement therapy, especially if the post-gene therapy environment is such that the factor level was on the low side. But by and large, the majority of the patients that one sees, and this is our experience, they do not need a replacement therapy. What about the safety profile of the currently available gene therapy? It is very elating, in fact, to see that the majority of the patients do not experience any serious adverse events. In the programs that I've just summarized, there were no development of de novo neutralizing antibodies against either factor 8 or factor 9. There were no deaths. There were, of course, a number of adverse events, the most common of which were infections. And in fact, part of the infections were as a result of the immunosuppressive therapies that we gave our patients because to manage transaminitis. And if one examines very closely the infections, the majority were of low grade, grade 1, very few were grade 3 and above. And if one looks at the relationship between the infections and gene therapy as assessed by the investigators, you can see that, in fact, the majority of these individuals felt that there was no relationship between the infections and gene therapy that the patients received. I just want to show you some of the data from the published studies to make 2 points. That despite the fact that the results of gene therapy are very, very much amazing, there is, of course, 2 factors that may well undermine those results. One is variability, and the second one is durability. And if we tackle variability first, you can see, as illustrated by this data from the SPARC 9001 study, in 10 patients that were enrolled in this study, they were given the same dose of 5E11 vector genomes per kilogram. What is shown here is a plasma factor 9 level as a function of time in days. And in fact, this study is ongoing. But I think what I want to illustrate here is that even though everyone got 5E11 vector genomes per kg, there is a fair amount of variabilities between individuals. We still don't understand why there is such variability. And my impression is part of that may well be the black box that I referred to earlier on, that we don't understand exactly what happens in that black box. What I also want to illustrate, in addition to the variability that is shown in this particular study, this is now the BioMarin 270 program, is the durability. All patients received 6E13 vector genomes per kg. As you can see, there were 2 dose cohorts that are shown here, cohort 3 and cohort 4. And of course, the variability is shown by the error bars, which are quite wide. But more importantly, is that when these patients were followed over time, on the left, you can see they were followed over a period of 3 years, and on the right, they were followed over a period of 2 years, that there seemed to be a drop in the factor 8 level as the patients progress. And of course, the big question we have is how durable is gene therapy in the context of hemophilia A? We don't see, of course, a similar drop in the previous slide that I showed you, which was on hemophilia B. So if one were to summarize gene therapy in the context of hemophilia, one would characterize it as a dichotomy of progress and challenges. the progress and opportunities that one can see here, you can see that in fact we do achieve therapeutic factor levels in a dose-dependent fashion. Of course, the higher the dose, the higher is the potential for toxicity. One can also achieve very low bleed rates, which is what we expect in someone who's undergone gene therapy. The ultimate goal in gene therapy is to have 0 bleeds in someone who's post-gene therapy. One can eliminate exogenous clotting factor replacement. As I indicated, there are a number of shortcomings associated with that. In the data that I've shown you and the data that is currently available, there are no safety signals, there are no de novo inhibitors, and there are no deaths associated with gene therapy in the hemophilias. The challenges, of course, that we face are many, and I've summarized a few here. We don't know, in fact, if gene therapy will work in the context of patients with chronic conditions. In the case of AAV-mediated gene therapy targeting the liver, we don't know if gene therapy will work if someone has got hepatitis A, B, or C, or HIV, because these patients, in fact, most of these patients were excluded from the studies. We don't know if you can re-administer. Common sense suggests that probably not, because as soon as the immune system is exposed to the vector, one develops immunity to the vector, make it highly unlikely that one can re-administer. Having said that, there are currently a number of studies exploring the possibility of re-administration in those individuals that may well need re-administration following the initial administration of gene therapy. We also don't know if it is safe in growing livers. These studies have not been conducted in pediatric patients. There is, of course, always a potential for insertional mutagenesis, which Dr. Koneta alluded to, and the choice of the vector, in fact, reflects that we do know that AAV-mediated gene therapy is expected not to lead to insertional mutagenesis. Of course, there is now non-human data that seem to suggest that these events do occur, even though the frequency of the occurrence is very, very low. Reason some patients develop transaminitis in those that have undergone hemophilia gene therapy, we don't know. In my own experience, in a very large number of patients that have undergone gene therapy, half of them have developed transaminitis and the other half have not developed transaminitis. And of course, you can't even predict when the transaminitis is going to happen. In some instances, it happens very early post-gene therapy, and in other instances, in fact, it happens very late in post-gene therapy. The good news, of course, is that it can be managed with immunosuppression, and secondly, to date, in our patients, the majority of whom have now crossed the second-year line, there is no evidence of transaminitis. It seems to be an event that happens very early in their lives. The durability is what I alluded to when I showed you the data from the BioMarin 270 program. And we also don't know, in fact, what factors are responsible for the variable expressivity that we see in gene therapy that I alluded to earlier on. What Dr. Koneta alluded to earlier on is the paucity of gene therapy programs in Africa. Perhaps to point out that 0.2%, in other words, 6 clinical trials have been documented to be conducted in Africa. We hope that with this webinar, we will encourage and coach and in fact make people to think about this picture. This picture clearly says a lot about us, that in fact we are not doing every effort to advantage our patients as it happens in other parts of the world, notwithstanding the fact that the resources may well be different. Ultimately, what we aim for is for our patients to have access to gene therapy, and that will depend on what product is available. Whether we do adopt gene therapy as part of the standard of care, whether we can use gene therapy appropriately, in other words, in the right patient, with the right dose at the right time, etc., etc. And perhaps what Dr. Koneta alluded to earlier on is whether we can afford it or not. And in fact, we're delighted that there are now programs that are beginning to address the question of affordability, particularly in parts of the world such as Africa, where clearly affordability may well be the biggest barrier that we face in rolling out gene therapy globally. That is all I wanted to share with you, and that is the end of my presentation. Thank you very much for your attention.
All right. Dr. Malangu, thank you very much. That was just a great talk and summarized many different things. I think we'll just remind folks to please, if you have questions, put them in the chat Q&A, because we will have an open discussion after the next talk. And so I'd like to welcome Dr. David Williams. He's a Leland Fix Professor of Pediatrics at Harvard Medical School. He's also Chief of Hem-Onc and the Senior Vice President and Chief Scientific Officer at the Boston Children's Hospital. Today, he's going to talk to us about really, I think, a very novel approach to the treatment for sickle cell disease. So, Dr. Williams, welcome.
Thank you, Ken. Thank you very much. I'm very happy to be here and share our thoughts about sickle cell disease treatment. And as Ken said, our approach is quite different than others. And I think the introduction that Ken and Dr. Malangu provided will allow me to skip through a few background slides. I would just note disclosures here as it relates to the current talk. I just point out that the vector construction that I'll talk about has been licensed to Bluebird Bio. And so there's a potential that will be developed as a clinical program in the future. So as mentioned both by Ken and by Johnny, you can think of gene therapy in two different spheres: direct application of the DNA contained in a viral vector or not in vivo, and secondly, taking the target cells out of the body, manipulating them using gene transfer, and then re-injecting them. I'm going to speak mostly about this on the left, but at the end, we'll come back to in vivo gene therapy. Now, I think that sickle cell disease has a global burden. It's one of the most common of genetic disorders. Worldwide, there's estimated to be 250,000 births per year. And in many parts of the world, children die at a very early age with this disease. There's about 100,000 patients in the United States, and here at Boston Children's, where I'm at, we care for about 300 patients with the disease. Now, there's multiple approaches to treating this disease currently being studied. And I won't spend a lot of time on these other than to mention them, but they're relevant in the sense of the fact that our approach is a little bit different. So one is to express in a viral vector either the gamma globin gene that doesn't sickle, or a mutated beta globin gene that's been generated that's resistant to polymerization of hemoglobin sickle and therefore sickling of the cells. The approach that we're taking is actually to target the repressor of gamma globin that inhibits the expression of gamma globin in adult red cells, and by this way induce elevated fetal hemoglobin expression. And then, of course, quite dramatically and with really nice early results, folks that are doing gene editing. One is to actually attack this target of gamma globin repression called BCL11A by removing an enhancer sequence that enhances the expression of this repressor in red cells. Another is to generate a mutation in the gamma locus called a hereditary persistence of fetal hemoglobin, and that also increases expression of gamma globin and therefore fetal hemoglobin. And then finally, of course, ultimately repairing the mutation that causes sickle cell itself, which is the position 6 glutamine to valine mutation. Now, why all this focus on fetal hemoglobin and gamma globin expression? So here is a simple diagram showing that in utero, as many of you know, you express primarily gamma globin, shown here on this red line, that pairs with alpha globin to give you fetal hemoglobin. And then once you're born, over a period of about a year, this gamma globin expression is repressed and adult beta globin is expressed. But of course, in patients with sickle cell, this adult beta globin is sickle mutated. And so when I see babies in my clinic, in hematology clinic, early in life, if they have sickle cell disease, while their fetal hemoglobin is quite high, they have absolutely no symptoms. And then once the fetal hemoglobin drops, because gamma expression drops and adult beta globin expression increases, they develop the typical protean symptoms of sickle cell disease. And I'd just like to emphasize that fetal hemoglobin is actually a very effective inhibitor of sickle hemoglobin polymer formation. So here's an old study, but it's quite informative. Here's a patient who has carrier or sickle trait. And here's a patient who has on one allele a sickle mutation, on the other allele has one of these hereditary persistence of fetal hemoglobin mutations. And so both these patients are completely asymptomatic from their disease. But what's interesting is that this patient with trait has about 40% sickle hemoglobin in his or her red cells. But this patient with HPFH and sickle has about 63%. And that difference shows you what a good inhibitor of polymerization fetal hemoglobin is. So there's been many years of study to try to understand that switch from fetal hemoglobin to adult hemoglobin that I showed in one of my initial slides. And this is work done by Vijay Sankran, who at the time was a medical student in Stu Orkin's lab. And he showed during development of red cells, during fetal development, this B-cell11A protein, particularly this XL form, gets induced as you get along in ontogeny. So in adults, you make a lot of this protein. And if he knocked down this protein, just look at this shRNA1 here, you can see in this column that there's a very big induction of gamma globin expression. And so this is really evidence that the B-cell11A protein is induced and represses the fetal hemoglobin, gamma globin expression during adult red cells. And so if you look at this cartoon, it shows you the critical role of B-cell11A in repressing gamma globin expression, and that allows the adult beta globin to express. So our approach then was to target this protein called B-cell11A using a lentivirus vector. So both Johnny and Ken have mentioned viral vectors. This is a lentivirus vector that we've had to engineer in a specific way. So the targeting sequence is shown here in red. And what we did, rather than express this as an shRNA, we engineered it into a microRNA, shown here. We call this a schmear. And this allows the cell to basically process this as an endogenous microRNA, which leads to less expression, so more physiologic expression and less toxicities. But importantly, it allows us to express this only in developing red cells, not in stem cells or B lymphocytes where there would be toxicity. And we do this using these sequences here called hypersensitive site 2 and 3, which are enhancers and hooked up to the minimal beta globin promoter. And this works quite well. So here you see a patient that's been treated using this vector, and these are lymphocytes from the bone marrow. And you can see there's very good expression both pre and post gene therapy of this protein that's essential for B cell development in B cells. But in the developing erythroid cells showed here, there's a very large reduction in the amount of the protein expressed in these developing erythroid cells in the bone marrow. So based on this and a lot of other preclinical data that I won't go through, we initiated a pilot and feasibility study for this first-in-human approach of a Schmier vector. And this was funded by the NIH and run by Erica Ezrick, who's shown here, who's a faculty in our hematology oncology program. And this just shows you the design. We generate stem cells in the peripheral blood by mobilizing them from the bone marrow using a drug called plerixafor. We select these target cells, CD34 cells, in the laboratory and use the lentivirus to transduce them. They're frozen until we do all the release criteria. Then the patient's admitted to the hospital for conditioning using a drug called busulfan. And once the busulfan it works and is out of the system, we infuse the gene-modified cells, and then we follow the patients originally for 2 years and then subsequently total of 15 years. So let me just show you a couple pieces of data from this study that is still open, almost done with its initial cohort of patients. So this shows you over time the presence of this Schmier vector in the blood cells of patients. And the take-home point is here's a number of patients treated, 6 of them, and you can see that there's a variability in how much of the gene is present in their peripheral blood cells. But on the other hand, the amount of vectors appears to be very stable over time. And what's more important is what this does with respect to the development of the red cells. So on the right, if we can start on the right, this is the percent of the red cells which have fetal hemoglobin in the blood. And you can see that this is quite remarkable because somewhere between around 70 to 75% of the red cells in all these patients contain fetal hemoglobin. And if we did this assay on me or you, if you don't have sickle cell disease, the number of F cells would probably be 1%. So this is a very large induction of the fetal cells. And you can see if we just measure the amount of fetal hemoglobin in the blood, it's quite remarkable. It's over 20% on almost all the patients. And the amount of fetal hemoglobin in these fetal cells is also quite high. This is quite important because this is the amount that's needed to inhibit the sickle hemoglobin that's present in the cells from polymerizing. And this dramatically is shown here. Here's the first patient that we treated. He's now over 3 years since treatment. This was a smear from when he was 4 years old. He's now 24 years old and has been followed at Children's all those years. Because of a stroke, he was put on monthly blood transfusions for 20-some years. And you can see all the irreversibly sickled cells in his smear. And here's his smear 6 months after gene therapy. And you can see there's no sickled cells. He's now, as I said, over 3 years off transfusions with a hemoglobin of 11 grams untransfused. And in that 3 years, he's had no painful episodes at all. He's had no chest syndrome. And this is true of the patients that we treated in this study. So this has been a very good treatment to reduce the pain. So almost all the patients have no pain. We've had one failure. This is a patient whose amount of vector manipulated cells in the blood is not high enough to prevent sickling, but otherwise the patients have been highly successful. It's interesting, one patient came into the study having monthly admissions to the hospital for a complication called priapism, and he's now, since 8 months, almost 2 years, had no admissions, no ED visits for this complication. And all the patients have clear improvement in their hemoglobin, not requiring any transfusion support anymore with some mild ongoing hemolysis. So this vector is safe in the number of subjects that we've treated to this point. And this really validates this as a molecular target that results in effective hemoglobin induction with F in a large number of red cells, that's important, with high amount of F per F cell, that's important, and a significant attenuation of sickling phenotype. So what we're doing with this study now is developing a multi-institutional phase 2 study that will serve as what's called a pivotal study for the FDA. The results could be used then to license it as a drug. And we had planned to open this this January. We've had to hold because of some complications, not in our trial, but in a different trial by Bluebird. But we're hoping to open this very quickly. And we have funding from both the National Institutes of Health in the United States and California Institute of Regenerative Medicine. We're trying to develop even better vectors. We would like to reduce the complications from the conditioning. So we and others are looking at trying to use immunoconjugates. And importantly, as I end my talk, we're trying to develop ways to do this by in vivo targeting. And we're doing this in 2 ways, using nanoparticles and new viral vectors. And this is a collaboration with the Koch Institute at MIT and MGH, and is funded by the Gates Foundation. So let me just show you that. The in vivo approach to sickle cell would be much more affordable, of course, portable, so we could do this in areas where the ex vivo manipulation of cells isn't possible from a GMP standpoint, doesn't require a special facility. And we would think that the stem cells that we're putting the gene into will be much healthier and happier by keeping them in the bone marrow or inside the body. And we ultimately hope that we can do this without significant chemotherapy conditioning. So one approach to this is using nanoparticles. Here's a scheme shown here where the nanoparticle containing the smear DNA is incorporated into a complex that has addressins or labeling targeting moieties on the outside to direct these cells to stem cells. So that's one approach. And the second approach is actually to adapt a different vector platform called an alpha retrovirus and to generate these in a way that has an envelope on them. This is the baboon envelope that directs these efficiently in vivo to hematopoietic stem cells. So this is work we're doing in collaboration with a talented junior faculty, Christian Brindle, in the gene therapy program at Boston Children's and Dana-Farber. And I'll just show you one set of data. So here are experiments where we've injected directly the virus with human CD34 cells into the bloodstream of mice that are immunodeficient. And over here you can see different conditions that we're using in these early experiments. So different amounts of virus that are tolerated versus cells only. And here you can see indeed in our best mouse, mouse number 1, we can detect at 4 months after transplantation, we can detect in vivo the transgene that we put into these animals by direct application. So that's very early data, but we hope will be applicable in the future to a wider variety of places where we could treat patients. And then finally, I'll just make the point that this gene editing is actually quite exciting. This uses data that was generated by Dan Bauer, who's a hematologist in our program, who showed that there's this little sequence here, enhancer sequence in the BCL11A intron, where if you cut this out, you now eliminate the expression of BCL11A in red cells. And so if this is done at high efficiency, this gene editing approach could equal the induction of fetal hemoglobin that we're seeing with the lentivirus approach. So this is sort of work that's currently going on in a company, and also we're getting ready to open a trial at Boston Children's. We hope these kinds of applications may very well have a broader application than the kinds of highly specialized centers like our own that are doing gene therapy currently. So overall, there's multiple new approaches to genetic treatment of sickle cell disease that appear effective to this point. Mostly these are ex vivo gene therapy methods, and they require extensive in vitro cell manipulations in critical care facilities. But a future goal will be to utilize the validated targets such as BCL11A and improve methods to translate these therapies into in vivo applications, which could be applied around the world. So just make one comment that I earlier alluded to, that our trial has been slowed down by leukemias that have developed in a different trial. This is the sickle cell trial called Lentiglobin gene therapy trial by Bluebird Bio. And this trial put ours on hold, although it's not clear the causes of this. It doesn't appear to be the virus itself. It appears to potentially be something about the makeup of sickle cell disease that may increase their risk for myeloid leukemias. So let me end there and just comment that this takes a huge effort. And I've mentioned Erica Esrick at Boston Children's already, but we've had support from our translational lab, the GMP cell processing lab at Dana-Farber. We have collaborators, of course, at Brigham and Women's and Mass General, but also collaborators in France, Bluebird themselves, and we've had funding by, as I mentioned, NIH and HLBI, Cure Sickle, and the Gates Foundation. And this is just a picture of the team. It's been developed over the last decade with a number of both program staff and faculty members shown here. And we have ongoing collaboration with a number of biotech companies and drug companies shown here. So let me stop there, and I appreciate the opportunity to talk to you about this exciting program that addresses a really difficult disease, sickle cell disease, that's a worldwide disease. And so we hope that this will be eventually be applied to many more patients.
Dave, thank you so much for a great talk, and we'll open it up now for questions. So please feel free to put some questions into the chat, and we'll, I think maybe just to start one off, you know, Johnny, maybe, you know, in terms of what's happening with your patients, do you see that gene therapy is of interest, and also just generally in the medical community there, what's the knowledge base around cell and gene therapy?
Thank you, Ken. And yeah, I've been quite surprised. And perhaps start with the second question. I think all of us, that includes some of us who have a little bit of experience, are actually in a steep learning curve. So the knowledge that we have, in fact, can obviously be improved through sessions such as this. But going to your first question, is gene therapy of interest to my patients? I was pleasantly surprised at the number of patients once we had opened our center for a number of gene therapy programs who were interested in the gene therapy programs. And of course, this was, some of it was surprising because some of these patients actually are doing extremely well on the therapies that I alluded to. The second, third, and of course the next generation replacement therapies. Quite clearly, over the years, we've converged with patients. We want to move towards a cure for a genetic disease. And I think that is one of the reasons why patients are interested. And I think the second reason is, in general, South African patients are very altruistic. They want to see things better than what they went through in general. And that is evidenced by the kind of, sort of participation in clinical trials that we conduct in Johannesburg, with over 90 clinical trials to date.
Yeah, well, thank you. And maybe I'll ask first of Dave, and then maybe Johnny, you can follow on for hemophilia. But for Dave, can you give a sense of when you see editing or other, particularly in vivo approaches, going to be available, and any guess of which will be the most successful?
I think, Ken, that what we need is a continuation of the sort of phase 2 clinical trials to generate enough data from enough different approaches to answer your question. My sort of guess is that there are going to be multiple approaches that will be effective. And so in the end, it'll probably come down to two things. One is side effects that are not anticipated until you do a large number of patients often. And the other is cost. And so those two things have to be worked out. With respect to in vivo delivery, you know, I think my feeling is it's likely going to be sort of on the 5-year to decade time frame. But on the one hand, you know, back in 1982, when I started on this, we thought gene therapy was going to be quick in the clinic, and it wasn't. And on the other hand, many things have accelerated much faster than I would have anticipated. So I think it's really hard to predict that answer.
So, Johnny, how about for hemophilia, what you're thinking?
I think along the same lines. I think David is absolutely right that perhaps the crunch is ultimately going to be balancing cost and toxicity, including genotoxicity, by the way, not just non-genotoxicity. But my impression, in as far as gene editing is concerned, within the genetic disorders, and specifically for hemophilia, I think we've got a long way to go. I think, in fact, it may be possible in the future, but given the complexity of, for example, the factor 8 gene in hemophilia with multiple mutations, it is not an ideal candidate for gene editing, where one, of course, has done the groundwork and has identified single mutations, etc. It may well be possible, but I think we've got a long way to go.
So, and Dave, maybe for the group, you've talked about conditioning, but many people might not know what the current conditioning is around that, and because I think that is a limitation for many reasons, and maybe just explain that a little bit more.
So traditionally, if a disease isn't a disease that has a tremendous selective pressure for corrected cells, even with the high efficiency that we can obtain with viral vectors, the number of cells you put back into a person is still small. And so to give them an advantage in general for most of diseases where you use ex vivo gene therapy, you give something to get rid of what's the residual stem cells in the body to give the new stem cells that you've manipulated an advantage. That's called conditioning, and it's used, many people know, for transplantation of many different kinds in the usual setting. In the gene therapy setting, it tends to be a chemotherapy agent. It often is a chemotherapy agent called busulfan, or busulfan in combination with other drugs. And so those patients get quite sick because of the lack of bone marrow that they experience while their graft is taking place, often for 4 to 5 weeks, and require intensive care hospitalization. So the clear goal for the future would be to eliminate the need for that kind of chemotherapy, but to use a conditioning that's much milder and could be, you know, potentially done, so the whole procedure could be done as an outpatient. That would be the goal eventually. I don't think we're there yet. There's some intriguing possibilities on the horizon, but I don't think yet we're seeing for sure those applications.
Right. We got one more question. We're a little over time, but I think Johnny, I'm going to throw one back to you, and that's I think a lot of the studies that you presented, were around hemophilia B, and there are reasons partly for that because the gene is much smaller and you can get it into AAV. Maybe could you just, as the last question, talk a little bit about hemophilia A, which is generally more common, and where you see that field going, because AAV is not the only vector people are exploring there.
Very important question, and I'll try to be brief because I mean that subject is extremely exciting. In part because we're not seeing the same results in hemophilia A when one compares to hemophilia B. But to answer your question, I think, in fact, as I've shown in the two tables that you have seen, the program seemed to be much more accelerated in hemophilia A, even though we know that hemophilia B actually was the first one to start. We are beginning to see the exploration of other vectors that may well be able to be used for hemophilia A gene therapy. And I think the biggest hurdle that we face at the moment, it is not clear in our own mind what kind of endpoint are we trying to achieve. Are we trying to make severe individuals to mild or moderate or normal? Depending on the answer to that question, it is possible that, in fact, one or more program may be able to address that, including some of the currently evolving programs.
So, all right. Well, one, I wanted to thank both speakers for really engaging talks. And let's go ahead and just cut our break time down to 10 minutes. So if we can reconvene at 20 after, wherever you are, we will we'll get to the next session. So again, thank you to the speakers, and we'll see everybody in about 10 minutes.
Hi, Dave. Are you set?
I just need you to stop the others sharing, and then I can carry on and open my.
I'm not aware of anyone who's sharing at the moment. Okay, that has just been done.
No, it was just me. It's just me. Okay.
It is 20 past. I would want to introduce the speakers for the next session. Gene and cell therapies are hand in glove, and this session is dedicated to the cell therapies. We've got an exciting program with two speakers, very experienced speakers. The first speaker is going to be Dr. David Britton, who is a clinical hematologist based at the Alberts Stem Cell Transplant Center in Gauteng. It's one of the few JC accredited centers, I think, in Africa. The second speaker is going to be Dr. Marcella Maus, who is an associate professor of medicine at Harvard Medical School. And her lab focuses on genetically modified CAR T cells as immunotherapies for cancer patients. And both of these speakers are extremely exciting indeed. With that, I would like to welcome Dave, and let's go ahead, please.
Well, thank you, Johnny, for that great introduction, and thank you to yourself and the American Society of Cell and Gene Therapy for the invitation to talk today. Can you just give me a thumbs up, Johnny, if you can hear me and you can see my slides? That's great. So, you know, what is the challenge of and what are the opportunities and challenges of cell and gene therapy for acute lymphoblastic leukemia and diffuse large B-cell lymphoma? I mean, this is, the whole world is talking about this. The transplant community is talking about this, and of course, all the hematological and oncological colleagues are talking about this. In South Africa, looking at the incidence of acute lymphoblastic leukemia, which is, although it's the most common childhood cancer, it does become more and more common as the population gets older. And looking at our population of about 60 million, we deal with about 600 cases per year, and about 400 of, based on the disease. In children, it's often cured by first-line chemotherapy in about 85% of people, but in adults, that number drops to around about 30%. So that looks like we're dealing with about 400 people who require salvage therapy, and about 200 of them, about half of those will either fail the salvage therapy or be ineligible for consolidation with an allogeneic bone marrow transplant. And the second diseases that we're talking about today is diffuse large B-cell lymphoma. And this is, again, quite a prevalent lymphoma, quite a prevalent malignancy, and again, increasing in age as we get older, and up to 90 per 100,000 population in people over the age of 65. In South Africa, and many other countries in the world, we see that it's much more common in people who are HIV positive, and this is due to multiple factors, but it's a reality. And in some centers, the numbers of patients that have been seen with diffuse large B-cell lymphoma has trebled. But what we're looking at overall is around about 3,000 cases per year, and about a third of them fail or are resistant to first-line therapy, meaning that about 800 people need salvage therapy. And again, only about half of those people have successful salvage therapy, and the rest are either fail or are ineligible for autologous stem cell transplant. So that means that there's a significant unmet need, but it's not massive. I mean, this is not in the sickle cell domain that we saw earlier, or even in in the other public health domains of HIV and TB. What is salvage therapy? It's usually a second line of intensive therapy using different drugs. It's often more intensive, it's often more expensive, and it's often more toxic, requiring hospitalization. And as one would expect, the more often or the more lines of therapy you fail, through a combination of selection and toxicity, one drives more and more resistance. And ultimately, patients need to have a bone marrow stem cell transplant to consolidate these salvage. And in the acute lymphoblastic leukemias, that's the allogeneic transplant. And in the autologous transplants are mostly used for the diffuse large B-cell setting. So I'll just put in a little thing about autologous stem cell transplant, just to give a bit of context to where we're going. And this is where we harvest the patient's own hematopoietic stem cells and cryopreserve them, and then treat the patients with myeloablative doses of chemotherapy or radiotherapy. And David Williams talked earlier about this kind of treatment as conditioning treatment. And it's in his context of the sickle cell patient, the objective is slightly different, but here the purpose is to eradicate the malignant cells, but unfortunately the collateral damage of the bone marrow needs to be rescued with the transplant. And the process requires a thawing and reinfusion of the stem cells after a reasonable washout period from the chemotherapy. An important point is that the patient's own cells are used and that they are unchanged or minimally manipulated in that setting. So what's with the CAR T cells? That's the genetic therapy that we're talking about in this context. And here the collection and the infusion process is similar, but once the cells are collected, they are genetically modified to express the chimeric antibody receptor. And the most common method of transfecting them is to use lentiviral vectors. There are some other methods in experimentation, but at the moment, the lentiviral vectors are the most commonly used and the most efficient, giving the highest percentage of transfection. And once the cells are transfected, they are selected for reinfusion. The vectors themselves are very carefully constructed, and I thought quite elegantly when I saw this with multiple plasmids that prevent them from integrating with wild-type viruses and sort of permitting wild-type viruses to carry their constructs and to do uncontrolled transfection. They're also carefully constructed to avoid known integration with oncogenes, and that is the safety profile has been demonstrated in a number of studies and also now over a protracted period of time. Once these cells are reinfused, they are the chimeric antibody receptor, antigen receptor, then brings them in contact with the malignant cells, and the T cells can then effectively kill them, and effectively they do. Just to give you an idea of the complexity of this process, you know, there's patient selection, and the patient in this situation is the donor as well. We've got this little cartoon, and the donor is cleared and undergoes the process of harvesting of their stem cells. And once that happens, and this distinction is important in the context of CAR T cells, and then it goes into the manufacturing process where they go through the processing laboratory, and the quality of the collected cells is established. They're transported, and at the moment it would not be a truck, it would be the volume is firstly too small, but it would be a plane, because there are only 3 regions in the world that are currently performing these therapies, being the United States, or rather North America, in a commercial way, Europe and China. The cells are transfected, and then these cells are returned to the treatment center where they're controlled and then infused back into the patient. Now, the complexity around this is important, especially when you live at the tip of Africa, and the logistics we will go into a little bit later again. So one of the first questions that arisen is, are CAR T cells autologous cells or a medicinal product? Because if they're CAR T cells, if they're autologous cells, well, that's pretty easy and everybody can carry on as normal. But if they're a medicinal product, then they have to be regulated as a medicine. But there's some problems related to that, in that because you're starting with different starting material, the different patients' autologous cells each time, you are, it's very difficult to standardize and assess them as a medicine. There are also some important points about once they are infused, is that the treatment themselves it expands in the patient. You once you give the CAR T cells, they expand dramatically, and so they're no longer the dose that you gave, and so controlling the dose is also not a matter of measuring what you give, but rather a matter of controlling what happens. They cause, in a significant number of patients, a cytokine release storm, which we're seeing, which has become popularized by COVID, and nobody asks you what does that mean anymore. But it is somewhat different in the transplant setting, or in the CAR T setting, and as such, it should be managed by people who have some experience in this regard. We talked about the logistic challenges, and just to highlight the issues here is that these are living cells. They don't have they have a finite lifespan unless they're cryopreserved, and if they're cryopreserved, that makes transporting them very difficult as well. And the patients that are getting these treatments, even if we were to move them earlier to what they currently have been used in, are still people who have highly aggressive diseases that rapidly expand. And the reason for using them is because these people have failed conventional therapy. So controlling the disease until one can get the appropriate therapy back to the patient is a medicinal challenge, and adding time to that and unpredictability is a problem. The patients also have to be lymphocyte depleted prior to infusion of the cells, and again, this is something that needs to be timed, and the logistics around that are just adds to the complexity of what you're trying to do. And given that you have a process that's happening ex vivo and a patient who's got a malignancy that's uncontrolled, this situation is problematic and often leads to a failure in the therapy because of the logistics. We mentioned that the product is different each time, and these are important points from a regulatory point of view. But once through the manufacturing process, one has to prove the transfection and prove that you have a satisfactory effector cell dose. And then once they're infused, you have to be able to prove, usually by flow cytometry on blood samples, that you've got significant expansion of the CAR T cells, and then by the same method prove persistence, and by measuring response to therapy in conventional ways prove efficacy. So to get to the topic of my talk, you guys thought I would be finished by now, and now I'm only starting. So the challenges are that we have this unmet need, and current availability of these treatments is limited by, and access already is limited to care for these patients because of the high cost of these medicines, of these advanced therapeutic medicinal products. We're going to discuss a little bit what that means and how do we use them. But some of the important things around this is the integrity of the chain of control from harvesting through to infusion of the cases. And we have to manage that by careful labeling and tracking of the product as it travels. And some of the things that I think every country in the world is starting to deal with are the sort of personal the privacy issues, and as I mentioned, the tracing and storage of these samples is logistically very complicated. So what we pointed out, though, is that patients who fail first-line therapy have a poor prognosis with salvage regimens. And even those that have a successful salvage regimen need consolidation with some form of stem cell transplant. And that salvage therapy on their own already takes a huge toll on the patient's health and the healthcare resources, and that each line of therapy that is failed further drives resistance and makes future lines of therapy less likely to succeed. I've included the EMA definition of advanced therapy medicinal products. They are medicines that are used in humans based on genes, tissues, or cells, and they offer groundbreaking new opportunities for the treatment of diseases and injuries. Ken discussed some of this stuff earlier, and he differentiated between germline and somatic cell therapies, and that this is something that anybody can Google. And just a point of reference here is that some ATMPs are incorporated into medical devices such as a biodegradable matrix or scaffold. Where we mostly concentrating on is in the CAR T cell situation is cells or tissues that have been manipulated or changed in their biological characteristics, or tissues not intended for use in the same essential function in the body, as in some of the induced pluripotential cells, where they forcibly change them to, say, for example, pancreatic beta islet cells. That would be an example of this. So clearly these products do need new rules. And is the current drug regulation process able to do that, since it is geared primarily to prove that a drug is in the compound, that the dose and the concentration is what we say it is, and a lot of the regulatory process is about the GMP compliance of the manufacturing process, and post marketing or through clinical trials, the toxicity and safety monitoring. But actually, is it really so different? And so I think there may be some difficulty in a regulatory system responding to new therapies, but the principles of why you have a regulatory authority in place remain the same. And so the mindset of the regulatory authority is actually appropriate, and it's adapting to the new product is not changed by its purpose, and because of the purpose, it's actually quite an easy change. So who has the experience and the systems to identify the suitable patients to clear them as donors, to harvest their cells, to manipulate, manufacture the ATMP, to prove their potency, sterility, and to administer them and care for the patient. And I think all around the world that process has been in the realm of the transplant physician, who are already doing a large part of this, except for the manufacturing and the quality control of the actual product. So what are the opportunities in South Africa? I think one of the things, I always remember my Prof. Barry Mendelow, who inspired me to a lot of this stuff. And many people in South Africa will remember him fondly. Not that he's not with us, he's just not teaching anymore. And he always used to say, you must watch out for statistics, because if you throw 10 balls up in the air and one of them stays up, statistically, that's not so significant, but it obviously is a very important finding. And in a group of patients who have got such poor prognosis and who have failed so many lines of therapy, the remarkable results that these products have had when treating patients has been akin to the ball that stayed up in the air. And so this high efficiency is important, and I think that we needed to take this and move it further up the line to treat people a little earlier than they've currently been done in clinical trials. But this has ensured rapid regulatory approval in North America and Europe to get these drugs, these cells approved. There's good scientific basis, there's rational application, and what we already have, as I was mentioning earlier, is we do have a sophisticated existing regulatory infrastructure. We do have a regulatory authority that is ethically positioned and scientifically capable of evaluating cell and gene therapies. And this operates in a sophisticated existing medical system. Yes, we have our problems, and we are resource limited, as Johnny has mentioned, in many ways. But we also have a healthcare system that is supported by excellent laboratory services, by effective logistics and distribution of medicines, and by integrated hospital and outpatient facilities. There's also further integration between the scientific community, the medical community, and the regulatory community. So I think that there's that this is a great opportunity for South Africa, is that we have a lot of stuff in place, and because of the scientific and rational evidence for these therapies, they are very attractive for us. This is a little bit more of the same stuff, but again, just pointing that out. How much time have I got? I'm just going a little bit over. I'll be quick. So we've got, we need to certify the apheresis and collection processes, transportation of the product, the manufacturing process is the one area that we don't actually have we have the manufacturing capabilities in South Africa, but we don't have this process certified yet. And that's where we need to get, where we need to move to. And then the management of the patients and the certification of this process is already in place. I just mentioned this is that HIV, we have a huge HIV burden, and although the antiviral therapies are very effective, there are patients who can't tolerate them, and there are a number of CAR T products and other stem cell gene therapies to convey resistance to HIV that may be applicable in this population of patients with lymphoma. So in summary, what I wanted to say is that although the implementation of advanced therapeutic medicinal products in South Africa is a regulatory and therapeutic challenge, it's also an opportunity for us to escape from the trap of increasingly futile and expensive salvage therapies. And starting with these two very specific disease entities with a very specific CAR-T therapy that is scientifically demonstrated efficacy is shown that it's not an insurmountable need. Our numbers are also not of the amount that would be. So although it's an important and significant, it's not in the millions or hundreds of thousands of patients. It's in the hundreds of patients, and it's an opportunity for us to enhance our regulatory, scientific, manufacturing, and clinical capacity, and to sharpen our tools in this specific field, so that we can leapfrog and enable us to provide care in the next range of cell and gene therapies for inherited, malignant, and genetic disorders. And with that, I thank you for your attention and again for the invitation for this. And sorry for going over time. I really didn't think I was going to.
Thank you very much, Dave, very much for that overview. And I think we will move on to the next speaker and then take the question and answer session. I would like to invite Dr. Maus, please, to share your presentation and go ahead. We can see your screen.
Great. Can you hear me now?
Yes, I can hear you.
Perfect. So thank you so much, Johnny and David and everyone for inviting me to speak as part of this global outreach from the American Society of Gene and Cell Therapy. I'm going to talk a little bit about developing gene-modified cell therapy. Some of it will overlap a bit with what David Britton just spoke about. These are my disclosures. So just to give you an overview of T-cell therapy and how it works in its current model, and I think this is still open to modification. This is, let's say, the first generation of gene-modified cell therapies. We're typically starting with a patient's own T-cell product, and so cells are collected through a leukapheresis procedure at the point of care, which is everything that happens up here in blue. And then their cells are shipped, that leukapheresis product is shipped to a manufacturing facility, which right now is being held mostly by commercial sponsors, but that it hasn't always been that way, and it doesn't always have to be that way. Where everything here down in yellow is the manufacturing center, and that is typically a center operating under good manufacturing processes that's very often FDA regulated in the US. So the leukapheresis product is stimulated with either antibodies or antibody-coated beads to enable the cells to enter the cell cycle, where they then become much more amenable to gene transfer, whether that's through a viral transduction or a non-viral transduction. And then the cells are expanded and cultured for approximately a week, and then the final cell product is harvested and formulated into infusible media, and it gets then shipped to the point of care where the patient is preconditioned with low-dose chemotherapy, and then the cells are infused into the vein in the patient to exert their therapeutic efficacy. There are multiple ways of engineering T cells to recognize tumor antigens. Typically, natural T cells will recognize antigens in the form of HLA plus peptides. So antigens are in the form of processed peptides that are presented in the context of the patient's own MHC. And most viral and tumor-specific neoantigens are actually intracellular proteins that would only be visible on the surface in the context of this HLA molecule. However, generating T cell receptors is a bit tricky. It can be done, but it's tricky to do at large scale because of the variability in HLA tissue types. In contrast, chimeric antigen receptors, or CARs, are independent of HLA, but the challenge is that they can only really recognize proteins on the surface of the cell that are still in their native conformation. And that's because the chimeric part of the antigen receptor here is based on an antibody binding domain. So it's taking the variable heavy and light chains and fusing them together into a single chain variable fragment, so that then the T cell will recognize antigen the way an antibody does. But the intracellular domains of the CAR will activate T cell activation, which includes cytotoxic effects, proliferation, cytokine production, and sometimes memory formation. And so chimeric antigen receptors are modular structures that can ultimately be used to change or modify T cell fate. And so they can be comprised of a binding domain. And what I just described to you was a single chain variable fragment, but this can also be done with single domain antibodies such as camelids or nanobodies. It can be natural ligands, it could be cytokines, or it could be peptides that are either synthetic or isolated from nature for their protein binding efficiency. There's also usually a hinge domain which separates the antigen binder from the transmembrane portion, a transmembrane domain, and then co-stimulatory molecules, which are listed here in orange, and then a primary activation molecule, which is shown here in red. And so the canonical chimeric antigen receptor is now composed of a single chain variable fragment fused to a hinge domain derived from CD8 or CD28, a transmembrane domain derived from either CD8 or CD28, a co-stimulation domain, typically CD28 or 4-1BB, and an activation domain such as CD3 zeta. But because these are modular, there's lots of different ways of testing and identifying new pathways for developing chimeric antigen receptors for particular diseases based on surface antigen expression. CARs can also, of course, be combined with other standard pharmacologic drugs that may enhance T cell function or may prime or sensitize tumor to T cell killing. And there's also additional genetic modifications that can be made to the T cells to either enhance their potency or enhance their persistence or otherwise modify their biologic behavior. There are now 5 FDA-approved chimeric antigen receptor T cells that are approved in the US. Axicell, or axicaptogen silolucil, is approved for, for large B-cell lymphoma. And then also recently, the same molecular construct, but with a different manufacturing process, Brexucell has been approved for mantle cell lymphoma. Tisagenlecleucel is approved for pediatric ALL and for lymphoma. Lysocell, or lisocabtagene maraleucel, or Breyanzi, is a defined composition, a 1-to-1 ratio of CD4 to CD8 T cells that is also approved for large cell lymphoma. All of these CD19-directed products, Axicell, Tisacell, Lysocell, and Brexucell, use the same antigen binder, so the same single-chain variable fragment, which is FMC63, a murine monoclonal antibody that is directed to the human CD19 protein. And so they're all potentially usable in multiple indications that are all derived from B-cell malignancies. They do have different toxicity profiles and different sort of kinetics based on the intracellular signaling domains, where we see a very sort of rapid on and rapid off rate with CD28-based CARs, and a much slower, slower kinetics of on, and then also prolonged persistence with the CARs that have 4-1BB signaling domains. Idacaptogene viclucil was most recently approved, or Abecma. This is directed towards B-cell maturation antigen in multiple myeloma, and it's now FDA approved, and it also has a 4-1BB signaling domain. And what's interesting about all of these chimeric antigen receptor T cells is that they've really changed the care of patients with hematologic malignancies in the US so far. And that's because the response rates observed and the durability of responses have really been unprecedented. So the first product that was approved, Tisacell, for pediatric relapse refractory ALL, in a highly refractory patient population, half of whom had already had an allogeneic bone marrow transplant, the overall response rate was 83%. AsGCT, when it was approved for large cell lymphoma, had an overall response rate of 72%. And the same product in follicular lymphoma has an overall response rate of 94%. These are just astronomical numbers, and that's been the case all around for all of the CAR T cells. If you compare it to sort of recursive chemotherapy, this is data from pediatric ALL, when clofarabine, an etoposide combined with Cytoxan was tested in 2011 in a phase 2 study. The median overall survival was 2.5 months. A biologic therapy like blinatumomab, which is an anti-CD19 bispecific T-cell engager molecule, is given as a continuous infusion, has a lot of initial toxicity, gets you an overall survival of 7.5 months in the pediatric relapsed refractory population. Very complicated to give. In contrast, CAR T-cell therapy, overall response rate of 81%, overall survival at 6 months of 90%, and median overall survival of 19.1 months. And so these therapies are highly effective in many patients, and then also in many patients, they seem to be durable. These are data from AsGCT. It's a CD28-based CAR in adult patients with relapsed refractory large cell lymphoma. And what's important here is that you see this flattening of the curve for both duration of response, overall survival, progression-free survival, indicating that even with this product, where the CAR T cells are short-lived, the responses are actually durable. And so one could make the argument that even as much as we talk about the resource intensity required for these therapies, in some ways, it's actually more of a one and done proposition where there's an intense time of manufacturing, monitoring, and so on that occurs over the course of about a month. But then afterwards, there's not this sort of recurring treatment regimens with recurrent toxicities for many years. So there are now data that has still actually been only in a press release, so I don't have slides to show you about it. But now these therapies, both AsGCT and T-cell, have been compared in randomized studies to autologous stem cell transplant, and it looks like these therapies actually extend progression-free survival and overall survival more so than autologous stem cell transplant. So I think, you know, the chances of these therapies moving up lines of therapy where patients overall will have less toxicity from multiple regimens and also lower cost to the healthcare system because you're not dealing with a patient who over years will be struggling against disease, but maybe at least half of the patients or about 40% of the patients have a chance of getting a durable complete remission from a single, albeit intensive regimen. So what about toxicities? The main toxicities with CAR T cells really revolve around 3 things. And these are the ones that are specific to the CAR T, because I did mention that there is lymphodepleting chemotherapy that is given ahead of time. These are typically lower doses of Cytoxan and fludarabine, but they can still cause transient neutropenia and infections. And so those toxicities are commensurate with chemotherapy dosing. But the ones that are specific to CAR T cells relate to how they function. And that is that when they get activated, they're biologically programmed to both kill tumor, which can cause tumor lysis syndrome, which is sort of can be a good problem to have. And then the second thing is that they release a lot of cytokines. And in that process, they can also activate the myeloid compartment, which then releases more cytokines, and then that can result in end organ damage, including hypotension, hypercoagulability, bleeding diathesis, so disseminated intravascular coagulation, and also temporary organ damage, including liver failure and sometimes renal toxicity. The primary mainstay of treatment for the cytokine release syndrome is tocilizumab, which is an anti-IL-6 receptor monoclonal antibody. Notably, IL-6 is mostly produced by the myeloid compartment, so we're not yet targeting the T-cell cytokines directly, but it's more the downstream consequences of that T-cell activation. The other significant toxicity, which is seen a little bit more with certain CAR T products than others, is neurologic toxicity. And the pathophysiology is not as clear as with cytokine release syndrome, but involves endothelial activation in the central nervous system and blood-brain barrier breakdown, which results in sort of a leaky and localized edema. And if it's very rapid onset and diffuse, then there can be cerebral edema and even herniation, but that is extremely rare. Most of the time, this endothelial activation and neurologic toxicity is a very transient phenomenon, and it's quite responsive to steroids. It can take a couple of days to weeks to get better completely, however, although we are now seeing studies where there doesn't seem to be any long-term repercussions of having had this transient neurologic toxicity. The third significant toxicity is B-cell aplasia, which is an on-target effect from targeting CD19. We don't see this with BCMA-targeted CAR T cells, it's only with CD19. And in adult patients, we generally just monitor, and if the patient starts to get recurrent infections, then we consider IV gamma globulin. But most of the time in adult patients, their plasma cell compartment and their antibodies are actually sufficient to maintain sort of vaccine titers like the tetanus and things like that. But we can, if we see recurrent sinopulmonary infections, we do give IVIG. The cytokine release syndrome, just to sort of go back in time a little bit, when it was first noted, was considered delayed because in contrast to drugs like rituximab, it's not, or cetuximab is not an immediate infusional cytokine release syndrome with fever or shaking, chills, or joint pains. It's a delayed cytokine release syndrome, and by delay, it can be anywhere from 12 hours to 7 to 10 days, and it's characterized invariably by fever and also by high production and high serum levels of interferon gamma, but most notably of interleukin-6. Which is how IL-6 receptor targeting was first tried in this patient population, and ultimately received an FDA label and indication for use of tocilizumab in managing cytokine release syndrome associated with immune effector cells. In terms of neurologic toxicity, there's been some correlative data performed, most notably by the Seattle group, showing that there is serum and CSF elevations in similar cytokines, and then also in additional antigens, including von Willebrand factor, angiopoietin-2, and other indicators of blood-brain barrier breakdown. In terms of the frequency of CRS and neurotoxicity with the FDA-approved products, these are the pivotal studies. There are changes depending on the product, such as whether it has a CD28 domain like Axicell and Brexucell versus a 4-1BB domain like Tisacel and Lysocell, and also can vary with regard to disease. So if you look just across diffuse large B-cell lymphoma, the 28Z-based CARs sort of behave the same, and the 4-1BB-based CARs kind of behave the same, where the 28Z-based CARs tend to cause a little bit more cytokine release syndrome, and about more than 40% of the patients require tocilizumab, whereas the 4-1BB-based tends to be less than that. In diffuse large B-cell lymphoma, more on the order of 14 to 23% of the patients requiring tocilizumab. ALL is a more difficult disease in terms of that the frequency of toxicity is higher, and it can be a bit harder to manage the cytokine release syndrome, particularly when the tumor burden is high in the bone marrow. One of the early challenges of the field is that everybody was using their own grading system for cytokine release syndrome. I think that has largely been solved now by having a consensus grading system that was published by a consensus group led by Trey Lee and Satwan Nilapu, and this was published in ASGCT 2019. And so this is now the standard in the field. The principles of management are based on grading of the cytokine release syndrome, grading of neurologic toxicity, which was also established by that group. And then for low grade is basically supportive care. And then neurologic toxicity it is managed primarily with steroids, whereas cytokine release syndrome is managed primarily with tocilizumab. And if both are concurrent, then both are given. The important thing to know is that tocilizumab does not actually improve or help neurologic toxicity, and can in fact even sometimes appear to make it worse. And so if there's a concern for neurologic toxicity, administration of steroids is imperative. You don't have to remember all of that. There's now an app that has been developed by the SITC Immunotherapy Immune Effector Cell Adverse Events Guidelines. This is also based on a paper that was published. I'm the lead author, and Steve Grupp was the last author, and it was also a consensus group, many of whom overlapped with the grading toxicities group. And so if you go to the immunotherapy guidelines on the SITC app, and you look at immune effector cell related adverse events guidelines, you can type in sort of check boxes for different aspects, like whether the patient awakens spontaneously. This is an example for the neurotoxicity grading. Whether they've had a seizure, And then it tells you you can click on the next link to go to the management algorithm, which will tell you sort of what steroids to use and for how long and things like that. I think there are many ways in development that are trying to be proactive about preventing neurologic toxicity. This is just one investigator-initiated protocol that we're running at my institution, where we are using prophylactic anakinra to block IL-1 receptor signaling in order to ideally, hopefully prevent both the neurologic toxicity and cytokine release syndrome in most patients in order to perhaps, or with the goal of enabling outpatient or at least less intensive inpatient hospitalization and management of toxicities. Too early yet to release data, so we'll see if the protocol is still open to accrual. In terms of challenges for CAR T cells in hematologic malignancies, even just staying within the CD19 and BCMA realm, I think it really breaks down into two, and David Britton talked about some of these. Some of these are scientific clinical and others are clinical logistics. And so the scientific clinical is that these CAR T cells are very highly potent and therefore can be difficult to manage toxicities, often requires inpatient hospitalization, namely due to cytokine release syndrome and neurologic toxicity, or ICANS. There can also be about half of the patients may relapse, and they do so about half of that time with antigen escape, and the other half due to lack of persistence of the T cells, where those cells just disappear too soon and the tumor comes back. In terms of future thinking, there's also the desire, of course, to develop additional and more tumor-specific targets to avoid toxicities and on-target effects. I think some of the major issues are also what I would consider clinical logistics. So right now it's about referral patterns to academic medical centers. It's difficult to administer these cells in community settings or in resource-constrained settings because of the potential need for intensive care. There's also a lot of logistics that just have to do with coordinating apheresis, cell processing, receipt of that process, paperwork associated with it for chain of custody, chain of identity, freezer logs for how the cells are kept, how they're thawed, et cetera. There's also all the regulatory compliance. So every cell pharmacy and every cell lab that is handling these cells has a pretty significant audit burden, both from sort of federal or professional societies like FACT, but then also sometimes from each individual sponsor. There's also a pretty high burden of data entry and staff training from floor nurses to research nurses to physician extenders to physicians themselves. And then there's the big elephant in the room, which is always about cost and reimbursement. This is a model that the U.S. healthcare system is already fragmented, and when it's covered by insurance and done as an outpatient, it can be a sustainable procedure. When it is not covered by insurance or is an inpatient procedure, it can be cost prohibitive. Having trouble advancing. Nevertheless, there is a tremendous growth in this field. If you look up chimeric antigen receptor publications in PubMed, you can see we're still in the log phase growth of the field. And if you look up CAR T trials on clinicaltrials.gov, we are also in log growth. And so what's happening is that there is a lot of new development of CAR T, and also individual countries that are developing their own CAR T cells outside of sort of the big pharma like Novartis, Gilead, BMS. And that's partly because the pathway to a gene and cell therapy clinical trial, which is outlined here, I would say is not exactly straightforward, but it has been published and it was first developed in academia. And so it really comprises of an innovation, at least in terms of IP, but if you're really trying to treat patients in the academic setting, it's not necessarily required. There has to be usually a set of preclinical studies that will identify the product and prove that it has some form of efficacy in a mouse model or in a preclinical model. A manufacturing process, and that's typically the most complicated and expensive in terms of maintaining sterility and processes around chain of identity and chain of custody. And then a clinical protocol and informed consent, as long as it's in the development pathway. In the US, those regulatory documents feed into federal agencies like the FDA, and then also local regulatory approvals like institutional review boards and safety review committees. I just wanted to briefly share with you that this can be done in academia. We identified CD37 as a potential target in mantle cell lymphoma, chronic lymphocytic leukemia, and also peripheral T cell lymphomas. And we identified first that there was homogeneous and consistent expression of CD37 across B-cell malignancies, and then in a fraction of T-cell lymphomas. And then we developed a CAR targeting CD37 and tested it in xenograft mouse models using cell lines and grafted into NSG mice, and then testing CAR19 versus CAR37 versus untransduced cells. And we're able to show that our two new CARs, that our CAR37 behaved similarly to CAR19, and that was highly effective compared to untransduced cells. And we did this in both xenograft cell line models and also in patient-derived xenograft models of mantle cell lymphoma and also of T-cell lymphoma. We then developed a process using the Clinimax Prodigy, which enables point-of-care manufacturing. It's an all-in-one enclosed unit, and I don't get anything from Ms. Honey for saying it. In fact, Miltenyi isn't the only one who makes this kind of instrument. There's actually now emerging instruments from Lonza and others to be able to manufacture CAR T cells either in bags or cocoons or Prodigy instruments, where sort of a self-contained, where you put on the leukapheresis product. You first, in the Miltenyi system, you isolate the T cells, you start them in the bioreactor culture system, add in vector, let them grow for 2 to 6 or 7 days, or up to 14 days, and then harvest those cells and formulate them in a final product. So this part of it theoretically does not have to be done in a GMP facility, although the portions around harvest and isolation should be done in sterile areas, and obviously with good documentation control and sterile practices. You can, in the US, we submit an IND to the FDA to allow us to proceed to a first in human study. And this consists of a regulatory sponsor, in this case me, and somebody who will write the IND and enable interactions with FDA, and then also immune monitoring, and of course a clinical PI who is not pictured here, who would enroll and treat the patients. And so I would say that it can be done. It's sort of a yes, we can scenario, and the hardest part is just setting up the manufacturing. But I think with the new technologies that are around, that will become eminently feasible. So CAR T cells, in conclusion, targeting CD19 have entered mainstream practice in academic medical centers in the US. These are highly bioactive, potentially curative therapy, which do require clinical infrastructure to be able to handle the consequences of giving patients these therapies. In the US, these are extensively regulated by FDA, FACT, and others, and that's mainly to ensure adequate training, reporting, and compliance for the commercial products. There is still rapid growth in our field at a global scale, but translation to trials is feasible even within academia, and financial toxicity is an ongoing issue, although I think different centers are resolving that in different ways. And with that, I'll thank you for your attention, and I'm happy to take questions.
Thank you very much, Dr. Maus, and thanks to Dr. Britton as well for this very comprehensive cover of the cellular therapy, the cell therapies at least. We have a couple of minutes, and I think we've caught up. We might not have as much time for the Q&A, but we've got a couple of minutes, and perhaps to kick that off, I just want to ask both of you, perhaps the first question. Given that the standard paradigm in cancer treatment is takes patients to first line, second line, and ultimately salvage, what do you think will be the impact of CAR T-cell therapy in this paradigm? Marcel, and then Dave.
Okay. So I do think that it adds more complication to the healthcare system to have multiple lines of therapy and then ending on a curative therapy or potentially curative therapy rather than the other way around. However, drug development sort of is designed to work in that direction, right? Where we first use established care regimens, and then when it's investigational, you sort of, when patients don't have that many other choices that they're known to cause benefit, you would start with an investigational agent. So I think we're right sort of at the cusp, because the approvals were obviously done after second line or more therapy, but that is changing. I mean, we don't have all of the data yet, we just have press releases, but even showing that these therapies are potentially second line agents rather than autologous stem cell transplant, I think that really changes the paradigm. And then the question will be, if eventually if they replace autologous or if they move up that line, what will the patients look like after relapse? Because for the half of the patients who do relapse afterwards, what will they be amenable to in terms of therapies, and will the disease respond? Because one thing that I sort of left out is that oftentimes the patients who relapse now after CAR T therapy don't necessarily do that well with other lines of therapy beyond that. But is that because of the CAR? I don't think anybody thinks so. Or is it because they've already had such multiply refractory disease and so much exposure to chemotherapy? And everybody thinks that it's that, but I think, time will tell as to whether that holds up if we actually move it up a line.
Thank you, Marcel. Dave, do you have a view here of the potential impact of CAR T cell? You have to unmute yourself, Dave.
So, of course, there are some other issues as well. The first thing is that if you use these CAR T cells in a very high tumor burden, that's when you get much more cytokine release syndrome. And the other is that when you try and harvest competent T cells from patients who've been repeatedly treated with lymphotoxic therapies, that may also have an impact. And so I think that Marcel is quite right, is that we're going to need to move up the ladder, but perhaps not quite to the front line. And the other question at the moment, as often is in new therapies, it's a one-off therapy, but there's no rationale or no, it's not irrational, I should rather say, to repeatedly dose patients. And the other thing that we're seeing is duo CAR. So when you have antigen escape, that's logarithmically more difficult if you have two targets. So I think that as these cells get more complex and less toxic, options like repeated therapies and preemptively managing escape will become easier.
Thank you very much, Dave. And I'm afraid we have run out of time in this session. It remains for me to thank both speakers for very comprehensive presentations. And I would now like to hand over to Dr. Cornetta to take over and lead the next session, please.
Thank you. Yes, that was a great session. Bring us up to speed on cell and CAR T therapy. And what we're going to now focus on is looking at regulatory considerations for early gene therapy trials. And it's been interesting on our committee. We had a conference previously with folks in Brazil, and we're, as I mentioned before, developing one with India. And again, folks who helped us formulate this said, what is a regulatory pathway in our country, and what is that going to look like for these novel agents? So I think it's an important thing that's important to make sure we are doing everything and not only trying to cure our patients, but doing it in a safe manner. So I'd like to introduce the first speaker, which is Dr. Paul Ruff. And Dr. Ruff is a professor and head at the Division of Medical Oncology in the Department of Medicine at the University of Witwatersrand, and also faculty in the health sciences. So he's going to talk to us today about health production regulation in South Africa. And Dr. Ruff, welcome.
Thank you very much, and thanks for the introduction. And thank you to Johnny for inviting me to present at this conference. I'm going to talk wearing two hats. One is the head of medical oncology at the University of Witwatersrand Faculty of Health Sciences, and the other as the chair of the Clinical Trials Committee at the South African Health Products Regulatory Authority, or SAPRA. And I just want to also thank Dr. James Southern, who I've mentioned here, who's a member of this committee, and he is a virologist and vaccinologist, but he also has a lot of insights into cell and gene therapy, and provided some of the slides that I've used. So I've just added him to the presentation. And so, as I say, I'm talking on behalf of the university and of SAPRA. Can we move on, please? Now, just as a very brief background of clinical studies, next slide. So when we review clinical studies, we see phase 1 studies, include first in human studies, and these are basically looking for dose-limiting toxicity and maximum tolerated doses for medicine. In cancer, it's unusual because we use patients who have no alternatives, unlike other areas of medicine where healthy volunteers are used, but you can't really use the type of treatments you use in cancers, be it solid tumor, hematological malignancies, in healthy volunteers. Then we move on to phase 2 studies, which may be single arm or randomized phase 2, and they're looking at adverse events and activity, usually the response rate, and they're not usually powered to show a survival benefit. And the main aim is to decide if the medicine warrants further testing in a phase 3 study. Next slide, please. And phase 3 studies are comparative studies. They're usually randomized. In the old days, there were some historical controls, and these are the most widely done studies, comparing a new therapy to a standard of care. Then also sometimes we review phase 4 studies, which are post-marketing, long-term follow-up with new therapies, especially when there are safety signals, and they're often mandated by regulatory authorities. Next slide, please. Now, this is the standard phase 1. I'm not going to go into much detail, where you do a dose escalation in 3 patients. If there's no dose-limiting toxicity, you move on to the next 3. If there is in 1 patient, you do 3 more. If 5 out of 6 patients are okay, you move on to the next dose level. Otherwise, you reach your dose-limiting toxicity, and 1 dose below will be a maximum tolerated dose. Next slide, please. Phase 2 studies are looking at new treatments. They're usually uncontrolled, open, one-armed and open label. And they're looking to see if there's a benefit for patients. They screen medicines for activity, and they extend the knowledge of side effects and toxicology and see if we can take these forward. Next slide, please. And phase 3 studies, which are the major studies that we review in the regulatory sort of environment. Next, please. And usually this is a study, and normally we see superiority studies, comparing a new medicine to a standard treatment, or a new medicine plus a standard treatment to a standard treatment. And you want to show if there is an improvement in the outcomes, looking at your relative risk reduction or hazard ratio. It usually should be less than 0.8. The confidence interval shouldn't cross 1, and you want a p-value less than 0.05. Next, please. We also look at non-inferiority studies, which will become quite important, especially when they want to see if a new medicine is easier or cheaper or something, and you're looking for a non-inferiority margin, usually 20%. So it doesn't show equivalence, but shows that the difference is less than a pre-specified margin. I said the usual is 20% with a hazard ratio of up to 1.2. Next, please. And this is just a sort of little cartoon showing how you compare the different studies from superiorities to inferiorities, whether you're crossing your 1 or you're crossing your delta of, in this case, 1.2, and making sure that your confidence intervals fall in the correct range. Next, please. Now, just with that as a background, I want to look at regulation of clinical studies in South Africa. Next slide, please. Now, regulatory authority is vested in SAPRA, South African Health Products Regulatory Authority. Was previously known as the Medicine Control Council until 31st of January 2018. And it's responsible for governance, quality assurance, and oversight of implementation of regulatory standards, procedures, and policies in clinical trials. It falls under the Amended Medicine-Related Substance Control Act, 101 of 1965, and the act that looks, the section of the act that looks at unregistered medicines in clinical trials is section 21, and the regulation for the conduct of clinical trials is under regulation 30. Now, as well as SAPRA, there's also an ethics component. All studies require ethics committee approval, and there are a number of ethics committees in the country, usually university-based or private practice-based, but they all fall under the National Health Research Ethics Council, which enforce ethics guidelines, policy, and promotion of good clinical practice. All studies are done under the South African Good Clinical Practice Guidelines of 2020. These have just been released about a week ago to replace the previous guidelines of 2006. They were a long time in coming. There was a lot of problems getting them released. Things were delayed a lot because of COVID and other problems, but finally they have been released. And they are very important for guiding studies in this country. And with the memory that although they do follow ASGCT and the most recent ASGCT from 2017, there are sometimes very specific areas that are important to this country. And South African SAGCP will supersede RCHGCP if there is a controversy. Although this isn't common, there are some country-specific issues that are put into the guidelines, and there are things that the people who work here know, things like post-trial access for treatment and the requirement for things like capacity building and transformation, but there are many others. And then also that's very important with clinical trials is that they all follow the ABPI guidelines, Association of British Pharmaceutical Industry Guidelines, most recent one of 2014, which provides for no-fault compensation for trial-related injuries. And this is very important, especially when you are doing early phase studies. Next slide, please. Now, this is just an organogram of what SAPRA looks like. Falls under the Minister of Health, but it's actually an independent body. It's a sort of parastatal body, has a board with a chair that looks after the governance and fiduciary aspects, and then a CEO who looks after all the actual in-house activities, and a number of expert committees. The most important one for us is the Clinical Trial Committee that looks at the clinical trials in new medicines, and the others that look at evaluation of new medicines. And also the other important thing which SAPRA does compared to the old Medicine Control Council is it looks at health products, not just medicines. So it also is involved in looking at medical devices, RVDs, in vitro devices, and radiation control. So it's much broader than just medicines in the past. Next, please. And here we have the just a little picture of the latest South African Good Clinical Practice Clinical Trial Guidelines, third edition of 2020, published from the Department of Health and under the auspices of SAPRA. And this was done as a joint effort between members of the Clinical Trial Committee at SAPRA and the National Health Research Ethics Council. We spent many, many weeks working together to finalize this. This document was actually finalized in 2019, but then COVID came along and delayed everything till now. Next, please. And then the ABPI guidelines are very important, and their basic principles read participant compensation, of who should be paid when participants suffer bodily injury and including death, when on the balance of probabilities, the injury is attributable to the administration of an investigational product under trial or any clinical intervention or procedure provided for by the protocol that would not have occurred but for the inclusion of the patient in the trial. It is regardless of whether the adverse reaction causing the injury was foreseeable or predictable, or the fact that the patient has freely consented, whether in writing or otherwise, to participate in the trial, and should be paid regardless of whether the patient is able to prove that the sponsor or applicant has been negligent in relation to research or development of the medicinal product under trial, or that the product is defective. Next slide, please. Now, SAPRA follows a global clinical research guidance framework. I mentioned RCHGCT, Declaration of Helsinki, the SIOM's ethical guidelines, and the Nuremberg Code. Next slide, please. Now, the Clinical Trials Committee that does all this work at SAPRA. Next, please. Next, please. Okay. So when a clinical trial is submitted, it's done on a document called the CTF1. This is submitted with the protocol and other documents, including investigator CVs, insurance, which is very important, workload documents of the investigators to see that investigators aren't doing too much, the ethics submissions, and then the declarations by the site and national principal investigators. And these are submitted, and the role of the national principal investigators become very important in clinical trials, especially during the COVID period, because they have an overall sort of view of what is going on in studies, as opposed to the site PIs who are only responsible for their sites. So CTC has 11 voting members and 5 external reviewers. It has expertise in most areas of medicine, including, amongst others, hematology, oncology, and now virology and vaccinology because of COVID, which is taking a lot of our time at the moment. We've reviewed up to 100 COVID studies in the last year. And these documents are sent to members or expert reviewers who carefully evaluate the submissions and write detailed reports for each meeting. Next slide, please. Next slide, please. Okay, so the reviewer presents, oh, sorry, go back one. The reviewer presents to the clinical trial committee, usually held every 2 months, although this has become a lot more frequent since the onset of COVID-19. We meet often weekly or even more often to try and keep up because of the urgency of the situation. We have to have at least 6 members to be quorate, and the decisions are voted upon by all voting members, and a score is sent to the applicant. The turnaround time used to be about 2 to 3 months, but is now a lot quicker since COVID came along. And if you have an amendment, these are submitted on a separate form called the CTF2, and this is not meeting dependent. It's done on an ongoing basis and should go through very quickly. Obviously, it depends on how complicated the amendment is. And if amendments are really complicated, we sometimes have to call for a resubmission. If there's a substantive change in a study, we cannot justify just an amendment and we need a new study. Next slide, please. And these are the scoring categories that we use. 1A is fully approved, 1B is approved but ethics pending, 2A is not approved but for approval by in-house evaluators, 2B is the most common, goes back to the original evaluators, and usually there's some small scientific issues that need to be done, and this doesn't delay the approval at all. Things get more difficult when you hit to category 3 because this has to come back to the full committee and it delays for another cycle. A 4 is unusual. It's when we have to refer for an outside opinion because there's no one who knows about the subject in the committee. Very rare, but it does happen. Number 5, basically it's not approved. And unfortunately we get a few of these studies. We've had quite a few during COVID, which are really just garbage and are thrown out because they have no scientific validity. And a 6 is because the administrative issues, that the applicant has not submitted the appropriate documents properly. Next, please. Now, with that as background, we're going to look at the regulatory and ethics issues related to cell and gene therapy studies. Next, please. Now, as a background, and most people know this, somatic cell therapy is defined as administration to humans of autologous, allogeneic, or xenogenic living non-germline cells which have been manipulated or processed ex vivo with the goal of treating or preventing disease. Gene therapy techniques include the use of modified human or animal viral or other vectors, including nanoparticles, now in the administration of genetic material, either ex vivo or in vivo. And the development of the CRISPR-Cas9 gene editing method allows for the rewriting and manipulating of a DNA sequence, either ex vivo or in vivo. Next, please. And it's just a little bit of background about CRISPR. The Cas9 enzyme recognizes a sequence stored within CRISPR patterns and cuts DNA. This is comes from prokaryotic species, which it is fatal to but in eukaryotes, it activates repair opening possibility to rewrite DNA. Adapting CRISPR from bacteria allows for editing of eukaryotic DNA, and you can cut DNA and activate a repair mechanism, allowing for potential rewriting of the DNA sequence, which sounds wonderful in theory, but is not always completely possible. Next, please. And this is just a little cartoon showing what's happening, how your enzyme cuts DNA and allows the insertion of a new sequence to basically fix a problem. Next, please. Now, you can modify the DNA of virtually any living species, and it's probably most viable in human diseases that are caused by mutations of a single gene. It's very difficult, if not impossible, to use this to change the gene in cancer where you have multiple mutations, but in diseases, especially inherited diseases, we have a single gene disorder, such as haemophilia, as Johnny knows. You could potentially cure this disease. And then you can do this either in ex vivo or in vivo. Next, please. But the problem with this concept is there are safety concerns. The Cas9 is normally present in bacteria that can infect humans, so our immune system is primed to neutralize it. An immune response to CRISPR or the vector could render the therapy ineffective, as well as inducing severe side effects. In the early 90s, everyone knows about the Jesse Gelsinger case, who died because of a massive immune response triggered by the adenoviral vector, and making that as possible that the ex vivo approach is safer than the in vivo approach. Next, please. Now, there are major ethical concerns, and this is from Professor Clem Perry, who's the chair of the ethics committee at our university. Is it ethical to modify the human genome? Initial applications of gene therapy aimed at curing genetic diseases seem straightforward. Genetic enhancement to give people traits beyond levels considered typical of adequate health may be of concern. At what point does gene therapy become a tool for eugenics? How should this technology be regulated? When should its use be allowed or banned? Germline editing research, i.e. sperm, eggs, or embryos involving hereditable DNA modifications, is of special concern because of the potential changes being inherited into perpetuity. Next, please. Now, in addition, there are many potential problem areas in cell and gene therapy which require regulatory oversight and hasn't been fully defined in this country. You have to define manufacturing procedures that ensure GMP, standards for collection of cellular materials and cellular starting materials, for reagents used to mature cell and gene therapy, standards for release of products for human usage, and sterility and freedom from adventitious agents and residues are critical prior to human use. Next, please. So standardized methods for determining potency and safety of these products are needed. Minimum standards for defining vector quality and safety for use in CGT is critical. And minimal standards for defining the quality and safety of gene sequences that may be inserted and or expressed in CGTs is critical. And the role of the regulatory authority, in this case, SAPRA, is critical in when these processes become more widely used, and it's an area that we're finding our way in. In addition, of course, the ethics committees also play an important role in regulating this type of research. Next, please. Next, please. Okay, now let's skip that one, it's fine. Now, one of the little problems we have is that certain parts of the of gene therapy regulation fall outside the Department of Health. Use of genetically modified organisms, or GMOs, fall under the so-called Department of Agriculture, Forestry and Fisheries, or DAF. Next slide, please. Now, the GMO Act number 53 of 1997 excluded human gene therapy from its remit. However, as this not is clearly defined, it's unclear how this should be regulated. And so we need policies to be developed for gene therapies that will include many of the concerns of the DAF GMO Directorate, but without its current agricultural bias, because really this is aimed at plants and not at humans. Next, please. But one of the requirements, and I'll just give an example here, is that if anyone wants to import any genetically modified material into the country, they need to actually advertise it in the media. This is an example of an advert that was placed in the media from for the import of a gene therapy, telemagene lahapavarec for multiple myeloma, and had to be advertised in the press to make sure that there were no problems with it and there were no complaints about it. This is an important requirement of DAF, but it sometimes goes against the ethics committee's requirements, and it's something that needs to be clarified. But anyone who's done any gene therapy or any studies in this country involving GMOs knows about this, and this advertisement is a legal requirement from the Department of Agriculture, Forestries and Fisheries. Next, please. And this is another example. Next, please. Now, there are further regulatory concerns about cell and gene therapy because we're using patient-specific product development. It precludes the accepted non-clinical and clinical trial development pathways for the production. In other words, like with the first gene therapy, angeloclusal CAR T cells, and many others that were mentioned by the previous speakers. Each product is designed and manufactured for a specific patient, and starting materials and reagents may not be identical. Therefore, it appears that the overall process should be regulated based on a defined set of input materials, resulting in a product that meets a specific standard for human use. Any regulations must be sufficiently broad to include all the currently known practices, as well as any technologies that are in development. Next slide, please. Research of these new technologies in South Africa and elsewhere has led to a number of promising discoveries, as has been mentioned before, that have benefit for patients short-term and long-term. And obviously people who develop these want to make profits. The lack of a clear regulatory framework has allowed some questionable CGT practices and procedures to become established in this country and in other parts of the world, including the USA. And you know, everyone knows about this sort of stem cell therapy that people propose is going to cure everything. And we've had a few problems in this country with people proposing stem cell therapy. And I'm aware of a lot of this happening in the USA. And, you know, we need proper regulation to prevent fly-by-nighters. And this needs to be done in a very strict regulatory control to prevent this type of thing happening. One of the problems is that specialists working in one field of medicine may not fully appreciate the complexities and risk of using these novel technologies without proper controls. And there are a lot of initiatives to integrate the responsible regulation of these technologies into the policy base of SAPRA. Next, please. Next, okay. And then lastly, just about it's also important to regulate the vectors. We need to have regulations to control the vector construction, characterization, production system, master viral banks, and lot-to-lot release testing and specification of vectors. And we have to have additional considerations for the different type of vectors that are used. Plasmid vectors, retroviral vectors, adenoviral vectors, and now nanoparticles. And we've seen a lot of this coming with the COVID, with all the COVID vaccines. And in fact, one of the benefits of COVID, I think, is going to help us further enhance gene therapy work because of the use. And we know that the different vectors have been used with viral vectors, nanoparticles, etc., for some of the COVID vaccines. So, you know, crises sometimes do have benefits as well. Next, please. When studies are evaluated of cell and gene therapy, preclinical studies have to define pharmacological and toxicological effects predictive of the human response in clinical studies. You have to look at starting doses, escalation schemes, target organs for toxicity, and which patient populations may have greater risk for toxicities of a given cellular or gene therapy. Next, please. Other important thing is the patients. So the PIL icon, patient information leaflet informed consent document, has to be specially prepared from an ethical point of view, and special reference must be made to the research uses involving genetic manipulation and human, non-human chimeras, as well as the exploration of donor objections to research. We need a national oversight research panel to facilitate this type of research in this country and in other countries. And then, of course, the issue of germline therapy and eugenics is something which, from an ethics point of view and from a scientific point of view, has to be regulated against, because those risks will always be there, even though we may not think so. So that the regulation of cell and gene therapy from a scientific regulatory point of view and an ethical point of view poses a lot of questions that need to be resolved and are being resolved in the development of this type of therapy worldwide and in South Africa in particular. And it's something which is very much a moving target in this country. We've started seeing some new gene therapy studies, especially from Johnny in haemophilia, and I think that we've got a long way to go in this area. Thank you very much.
Professor Ruff, thank you very, very much. It's, you know, gene therapy is complicated, but regulations, whole nother level. So thanks for trying to get everyone to understand that. And, you know, and just to hear how you're formulating it and try and bringing things forward, and both trying to bring things forward, but also protecting patients. So I'd like to introduce our next speaker, Adora Ndu. She's both has a doctor of pharmacy and a law degree. And so has been very involved with BioMarin Pharmaceutical and looking at their regulatory, but has also been the chair of the ASGCT Regulatory Affairs Committee. So we're looking forward to hear her talk and give us her perspective on regulatory considerations for gene therapy development in South Africa. So welcome.
Thank you. I'm just confirming that you can hear me and that you can see my slides.
Good.
Great, thank you. I'm really thrilled to be here today. It's an exciting time with gene therapy development, a lot of activity, and evolving regulatory landscapes globally. So my goal today will be to share some perspectives focused primarily on the US and EU, and to help drive conversation around similarities, differences, opportunities across the board. So here's my statement, which essentially says that the views that I share today are mine. But before I dive into my content, I do want to share some background into what we're doing at BioMarin. Currently, I lead our worldwide research and development strategy, scientific collaborations, and policy at BioMarin. And we as an organization are focused on translating genetic discoveries into transformative medicines. And so this just gives you a snapshot of our portfolio, which includes 6 commercialized products. But I'll just draw your attention to the bottom just to highlight our areas of focus for gene therapy as far as what we've disclosed publicly. And as you can see, we have a number of gene therapy programs that are advancing Haemophilia A, PKU, hereditary angioedema, and HCM as well. And so our experiences are through different stages of development, varying interactions with health authorities globally. So I hope to share at least some points from a regulatory perspective today. So just to start on a little bit of background, I actually ended up removing all the background information specifically about gene therapy because the earlier speakers have done a really phenomenal job going into the deep dive about the different types of products. But just to give you a little snapshot of history and where we've been and where we are now, and I'll touch on where we hope to go. But you know, we have a long history in this space as a community, dating back to the early 70s. I was able to identify this paper that was published in the Science Journal about the possibility of gene therapy and what it potentially means for human genetic diseases. First clinical trial was kicked off in 1990. And you know, a lot of other activities since then, the Human Genome Project, the unfortunate death of Jesse Gelsinger, which was mentioned earlier, following serious side effects after receiving a gene therapy with the adenovirus vector. And since then, the field was set back for quite some time. I think there was a need to really understand the safety of gene therapy products, you know, really improve the technology around the vectors and establish some frameworks that can ensure patient safety in clinical trials. And so since then, at least the first gene therapy product that was approved was actually approved in China for head and neck cancer. And I've kind of dropped in some regulatory advances here. So you'll see 2007, EU published their regulation on advanced therapies. ICH has also issued some guidelines on various topics, and I'll highlight those a little bit more later. And if you go to the red box at the bottom, I've also tried to flag some more recent activities, including some approvals. EMA's first gene therapy approval for Glybera, which was mentioned earlier, unfortunately only treated one patient, so didn't do very well in the commercial setting. Japan has also been very active in this space. They have a legislative framework, the PMD Act, that supports development of gene therapies in Japan. US has also done quite a bit to advance their regulatory framework, more recently in 2016, the 21st Century Cures Act, which introduced the RMAT designation, the Regenerative Medicines Advanced Therapies. And then you see some recent approvals dating back to, in the US, dating back to 2017. Some of these have already been mentioned. So really rich and active history across the board to advance the regulatory frameworks to support actually getting these transformative products to patients and getting patients access to these. So as far as the current state, as I mentioned, there's really been a spike in activity. If you ever listen to an FDA talk, they always highlight the significant increase in INDs, IND applications that are submitted for gene therapy products. And this is a snapshot just from clinicaltrials.gov. And the numbers can vary depending on, you know, which date you run your search. But as you can see, this is a global development effort. There are clinical trials all over the world. You know, some in more areas than others. But the takeaway here really is that this will continue to increase as more researchers initiate trials in different countries. And there's certainly opportunities to increase in some areas like Africa. You know, so I'm hoping to see that increase as we move forward. And at the bottom left, you'll see the general areas of focus. There's a lot of activity around oncology, cardiovascular and CNS, infectious disease, metabolic, autoimmune, and ophthalmology. And so this is another snapshot of some select global approvals. And the reason I say select is because this can literally change by the day. But these are some that are highlighted here. You'll see that most of the approvals that we have to date are really in the US and also in the EU, followed by Canada and more activity in other regions. So we hope to see more of these as we move forward and bring these treatments into clinic and go through the regulatory process to actually get these products approved. We hope that by next year and subsequently, there will be so many approvals that we wouldn't even be able to actually name these products on the screen. So really where we are today is we know that there's an unmet need. We know that most gene therapy products are being developed for rare diseases, which is an important point that introduces its own suite of challenges. But we know that there are unmet needs for patients, and that's why it's so important for us to collaborate, academic researchers, industry, health authorities, to really establish the infrastructure, the framework that will support development of gene therapy products and support the ability for these products to actually go through the regulatory hurdles to get approved for the benefit of patients. And we know that the science and the technology is evolving at a speed that's actually much faster than our frameworks are able to keep up. And so as we look across different regions, the question that comes up typically, which is an important one, is how can we better converge? How can we better harmonize to ensure that we are able to keep pace and we are able to actually support development of these products moving forward? So just to take a little bit of a deeper dive into some of the frameworks, I'm going to focus primarily on the US and the EU. We've heard in the previous presentation about South Africa, but this will just give you a little bit of a context. So I've highlighted here just some select regulations and guidelines. There are many more than this, but this gives you a sense. ICH has over the years increased their activity and their focus on the guidelines that are needed. But as we know, the ICH process is quite lengthy. It takes some time. There are a number of countries and regions involved. The US and EU have also established regulatory frameworks to support the development of advanced therapies. So in the US, a number of regulations, more recently, several guidances have been issued focused specifically on gene therapies. You'll see some listed here. CMC for gene therapies, human gene therapy for rare diseases. As I mentioned earlier, we see many of these products being developed in the rare disease space. So these guidances try to provide some clarification to researchers and to industry developers on how to develop these products: clinical trial design, preclinical considerations, statistical considerations, long-term follow-up, safety, et cetera. And similarly in Europe as well. So these are just some highlights of the US framework. And as I mentioned, more recently, if you look at the bottom here, more recently, FDA has issued a number of guidances. Some of them have been finalized. And these are, I would say, a good reference point for other regions that are still developing their regulatory framework. I know I can say that some countries in Latin America have kind of looked to what US and the EU are doing and incorporated some aspects, because ideally, to help support development, ideally we want to make sure that what's being recommended is not deviating significantly, because it just raises the bar. It creates more of a barrier, I would say, for introducing products in as many countries as possible. And in the US, gene therapy products are reviewed and regulated by the Office for Tissues and Advanced Therapies, which is in the Center for Biologics Evaluation and Research. And I included the gene therapy definition here because I think one thing we've also found is that the definitions can differ from region to region. And this introduces its own complexities when what is considered a gene therapy is different, because that also dictates what regulations would apply to those products. So I think this is an opportunity area for us from a regulatory perspective, is to really determine and align on what the definition for gene therapy is. And in the EU, I've included some really important documents here, the directives, regulations, and guidelines that are specifically for ATMPs, advanced therapeutics. And as far as the regulatory oversight, the structure is quite unique in that we have the Committee for Advanced Therapies, which provides the scientific opinions on cell and gene therapy products specifically, and they work closely with the CHMP, the Committee for Human Medicinal Products, in providing that scientific opinion, and CHMP ultimately, in conjunction with the CAT, issues the opinions. And they also have an innovative task force, which is available for support for developers in the earlier stages. And here you'll see the gene therapy definition, a little bit different from what we have in the US. You know, I think ultimately they for the most part align, but you know, there are nuances that do make them somewhat different. Also, another aspect of the regulatory framework that is important are the incentives or the programs that support expedited development. And so starting with the US first, there are a number of programs that are available for researchers and developers to leverage, to apply to. And if your product is addressing an unmet need or treating serious or life-threatening conditions, and these pathways all help facilitate and expedite development. So starting with orphan drug designation earlier, fast track, breakthrough, and RMAT all provide opportunities for closer interactions, expedited reviews. RMAT is right here at the bottom, the Regenerative Medicines Advanced Therapies. This was actually created specifically for regenerative medicines, which includes gene therapy. So it's quite similar to this breakthrough designation, and it provides an opportunity for developers to have regular interactions with the agency to receive advice on their products to put, move them forward, and also provides an opportunity to qualify for the accelerated approval pathway, which allows earlier approval based on a surrogate endpoint. We also have priority review as well. So, and at a later slide, I'll show you a snapshot of some of the more recent approvals and which of these designations they were able to obtain. And then similarly, in the EU, EMA has their own framework to help expedite development. So again, orphan drug designation, they have the Innovative Task Force, PRIME, which provides the opportunity for that continued interactions. Conditional approval, accelerated assessment decreases the time frame for review, and also exceptional circumstances. So I think the takeaway from these two slides is really just the importance of having these programs in place to support development, to support getting that close collaboration, that advice that's so critical for a space where the science and technology is evolving very quickly. So those close interactions are important so that these, researchers, developers can get the advice needed to establish the right, clinical development programs to, move things forward to ensure safety of the patients that are the subjects that are included in the trials. So this is, again, just a snapshot of some of the recent approvals, and this is just focused on the US and EU. The products are all listed in this first column, and at the top you see some of the designations that I mentioned in the last two slides. And so it's interesting when you look across this particular slide, how many of these recently approved products were able to leverage some of these designations to help support actually developing and getting these treatments to patients. And so it's, again, really important to have these frameworks in place. So just a few development considerations, at least from the regulatory lens, that I wanted to walk through. As I mentioned earlier, we see that many of these products are being developed for rare diseases, and rare disease drug development does present its own unique challenges. Some of the challenges I've listed below include the small size of the populations. We know that each region has their own definition of what a rare disease is, but it presents some challenges because the typical large placebo-controlled randomized trials may not be feasible, and so that's something to consider. That's something to ensure that you're speaking with the regulators about on a regular basis. And then patients are spread out. So there's a need for more centers, multi-center trials in order to identify patients and enroll patients in the studies. The natural history is typically poorly understood. There's phenotypic and genotypic diversity. So you have a variable disease course. And there's typically a lack of validated endpoints in some of these areas where the condition is so rare and there are no approved products. You're typically charting that path. And so again, another opportunity and really important for that close collaboration and communication with the regulators. And there's lack of precedent. So you're really paving the way here. As I mentioned already, we have smaller populations when you're looking at rare diseases. And so the evidence that's required to support approval of a gene therapy for a rare disease is something that it's really critical to ensure alignment with regulators on that. And typically sometimes what you see is one adequate and well-controlled trial has been acceptable, but again, you know, these are points that really need to be aligned up front. A few points here with regards to GMOs, which, you know, depending on which country or region, you know, the requirements may differ. And so that can be challenging for researchers and developers that are trying to navigate, one, how GMOs are defined, and then based on the definitions, how to navigate the regulatory path, the requirements, the process, who needs to approve what. And so this is an area that I know that at least the industry stakeholders have been really looking forward to the opportunity for more alignment across the board, just to help streamline the process for GMOs. So, you know, really important to have clear guidelines and approaches to support approval. And, you know, some GMO frameworks are a bit dated, and, you know, so there's a need to revisit some of these frameworks to better streamline and also ensuring that there are global standards that are feasible across the board. A few CMC considerations that I will go through these very quickly. I'm not a CMC expert, but what we do know is that CMC is one of the most important aspects of development and can actually delay development and approval of products. And so from a global perspective, one of the important things is really to take an approach of looking at where we can harmonize, where we can converge on some of the specifications for development and approval. Leveraging the possibility of a common CMC dossier will help streamline the process. Again, I can't emphasize enough the importance of communication between the agency and the developer. And, you know, some of the challenges with gene therapy specifically, possibly needing to use fewer lots, manufactured in a shorter duration, and the limited data that would be available for establishing the specification criteria. A few points from a global perspective that are important to us and has been, I know, a focus from ASGCT. We just recently issued a publication on this, actually, on the importance of concepts such as regulatory reliance and work sharing. And so I think this is important for this particular context, given the global forum. But, you know, I can't, you know, overstate the importance of leveraging reliance when feasible, because health authorities are under a lot of pressure to, you know, improve performance, to facilitate timely access, and to have, you know, processes in place to support regulatory review and approval. And we know that while some countries and regions have made a lot of progress in establishing the regulatory framework to support review and approval, you know, others are still very early in that process and will take some time to catch up. And so according to the WHO, reliance is a concept where one regulatory authority will take into account and give significant weight to the work, the review work performed by another regulator or another trusted institution to reach its decision. And so you see this across, you know, different countries, leveraging reliance activities during review when, say, if FDA approves a product or if the EMA approves a product, really taking the approach of determining how much to rely on that review and approval to help decrease the duplication of work, increase your regulatory capacity, improve regulatory efficiencies, and support faster approvals to make these products available to patients. So that's a concept that there's a lot of energy around that because of the possibilities that it brings. And then the work sharing is also an approach from a regulatory perspective that can increase efficiencies and really focuses on that international collaboration, again, to decrease duplication and redundancy. And there are a few examples of this, and one that I included here at the bottom is the Access Consortium, which is a work sharing agreement between Australia, Canada, Singapore, Switzerland, and the UK. And essentially they take an approach to work together to collaborate in their review, and they divide the regulatory workload. So, you know, a certain country might take ownership of a certain module and review it regionally, but ultimately, and put forward a decision. Ultimately, each agency is still responsible for their own final decision, but they leverage the work that's been done by another health authority. So these are just concepts to consider. And ultimately, the key takeaways are, again, as I started, it's a lot of activity in gene therapy, many clinical trials underway. We have several new products approved. And so it's here, and the work will increase. And I see this as an opportunity, you know, ties closely to my second point, which is about patient centricity. Many of these products being developed for rare diseases, so there's a significant unmet need, and safety is critical. So taking a patient-centric approach is really important. Global alignment and harmonization, I can't, you know, overstate the importance of why it's important for us to help facilitate that global clinical development. And then we need the science-based policies developed. We need a regulatory landscape, and as well as expedited pathways, so that these, you know, that can allow for closer interactions with regulators to support developing and bringing these products to patients. And then again, regulatory reliance and work share, always two key concepts that are critical in the global landscape, especially because we know that the science is evolving very quickly, the technology is evolving very quickly. So being able to collaborate across different regions to help support the work will be important. And that's my last slide. So thank you.
Wonderful. Thank you so much. Dora, we'll just open it up for a couple of questions. I guess one thing, as I was listening to you know, your talk, and you're talking about expedited pathways, and particularly those have been used often in rare diseases. And while we say rare diseases, if you think the number of rare diseases is huge, and often they really can be very intensive in the cost of the care for these individuals. So I think part of that's been the impetus, but maybe just to get your opinions, because I do think, at least in the US, part of the reason the FDA started to develop some of these pathways was not only because of physicians trying to push it, but I think patient advocacy groups have played a role in that too. And maybe just, Adora, do you think that that's a true statement? And Paul, maybe to think about advocacy groups in South Africa and what role they play potentially in cell or gene therapy.
Yeah, no, you're absolutely right. Patient advocacy groups are a critical part of any development program. And if they're not, they should be. Because we know that patients are the experts in their disease. They live, they breathe it every day. And so what we see in the US and Europe especially is for researchers, close collaboration with patients and advocacy groups, even as early as the preclinical stage, as early as developing the clinical development program, to get some insight and feedback into the design, the protocol, and that again, trying to understand what are the outcomes that are most important to patients. And so from the expedited programs perspective, FDA, I know, has collaborated very closely with advocacy groups. And it actually, a lot of this started in the oncology and HIV space, where advocacy groups were really encouraging the agencies to think about ways to help expedite the development of these products to bring them to patients, and so that patients can have the access. So yes, you're absolutely right.
Paul?
Actually, the issue of rare diseases or orphan indications is a very vexed issue in this country. We've had a lot of discussion about what to do, because when you are resource constrained, can you afford to spend large amounts of money on a small number of patients? And this is an ongoing debate. I mean, this has been going on, you know, with storage diseases. It's even with haemophilia, as Johnny knows. It's getting more and more complicated. Is how can a low middle income country afford to provide treatment, even though it may be curative, to you know, rare diseases? And the treatments will never become affordable because the numbers are too small. And so it is something where we don't have a policy, and in fact we've been trying to develop one, but we're having a lot of difficulty in it. You know, you do have advantages in the US and Europe with tax rebates and things like that, but here it's very difficult. So this orphan disease story is a huge problem, which I think is going to be very difficult to resolve. And as it gets more and more complex, as Adora mentioned, I think it's going to get more difficult, not less difficult, actually, to provide these type of therapies. And you know, the issue about advocacy groups or activist groups, we have a very strong community in this country. We've got a group called the Cancer Alliance, who are very, very potent, and they're a group of about 30 different advocacy groups, and they're very effective in lobbying the health department and the government and that when it comes to providing medicines. But, you know, there are limits, and the limits become, ultimately the limits we have are financial. And, you know, and you're in a country that's got a, you know, a massive outbreak of HIV and TB, and now with COVID as well, the ability to provide care for these costly medicines is getting worse and worse. And I mean, I think even that's affecting everyone. I mean, one of the problems with COVID, and it's something that we've had a lot of discussions and even published on, is that the effect of COVID is on people without COVID is much worse because of the negative impact on overall health care. Although maybe there's a light and, you know, there's a silver lining, as I mentioned. The research developed in the development of COVID vaccines is going to help therapies going forward. And then with all these novel vectors and other approaches is going to obviously have applicability outside COVID vaccination. So it's a very difficult area. And I think in this country, it's something which I don't see us really resolving in the near future. Thanks.
I want to be, you know, conscious of the time, and I'm for sure a lot of folks are getting hungry on here for dinner. Professor Ruff, there is a question in the Q&A for you, if you wouldn't mind trying to answer that. But right now I'll turn it over back to Dr. Mlangu, who's going to finish out the meeting.
Yeah, you mean REC, Research Ethics Committee? Is that what's meant?
Yeah, you can. I think you can type your answer there into the.
Yeah. Oh, should I type an answer there? Rather, let me answer live. This is in this country, all research requires approval by both research ethics committees and SAPRA, as I mentioned. The most sort of important ethics committees are the university-based ones, and they deal with them. We don't have a specific ethics committee that would approve cell and gene therapy, but you know, all clinical research, no matter what it is, would go through the SAPRA Clinical Trial Committee and one of the ethics committees. There's a little problem because the university ones are very stringent and do cover academic work, but you know, there's a lot of people in the private sector doing research as well, and very good research, and the ethics stringency is not quite as good as it should be. But at the moment, the ethics committees would provide approvals for any form of clinical study involving any sort where humans are involved. That obviously that maybe changes, but that's where we stand at the moment. Thanks.
Thank you very much, Paul, and thanks, Ken. It remains for me to just do a quick wrap up, and perhaps the noteworthy thing is that collaboration and partnerships are key to progress in medicine, and we're very grateful for this collaboration and partnership with the American Society of Gene and Cell Therapy, which has partnered with a number of South African medical organizations and individuals to bring this education webinar to Africa. We hope this is not the last, but the beginning. What I also heard very loud from this webinar is that the ASGCT has these global partners in all continents around the world. And I think that, in fact, it is the right vehicle for us to get on in order to be able to get the expertise and the skills and the knowledge that we need to progress the gene and the cell therapy fields. I consider today's webinar a huge success by all counts. However, there is a need for us to perhaps explore other focus areas such as, for example, the ethics of cell and gene therapy, the funding models, manufacturing groups, creating more partnerships. I think those are some of the areas that may well be explored in the future. And finally, I'd like to say a particular thank you to Sam, Betsy, Caitlin, and Ken of the ASGCT outreach team for taking care of all the logistics culminating in this meeting. And finally, thank you to all the attendees, including all of you who have hung around until the end. Perhaps to improve the next seminar, I would like to request that you please complete the survey that is going to appear in the chat so that we can deliver a better webinar when we meet again. With that, thank you very much and have a good evening.
Can I just say one thing before we go? Just to thank Johnny for all the efforts he's put in organizing it, and thanks for inviting us to present. And just to say, the only way to do this type of thing is to collaborate. It's a very difficult area. There's lots of questions. I think from this meeting, there's more questions than answers, but I think at least we now have questions, and I think that collaboration is needed at a national level in this country and at an international level, because this type of approach is very exciting, but there are many difficulties, and it's going to take a lot of work from a lot of people, and the more we work together, the better. So thanks, Johnny.
Yes, thank you.
And thank you, everyone, and all the speakers as well.
Thank you, everybody. Have a good night or a good day.